Our next session is about to begin. May I request you to kindly mute your microphones and wait as we prepare for the session. Thank you.
All right, good morning, everyone. Welcome to Professor Inti Banerjee Memorial International Conference on Translational Research in Medicine, TRIM 2022. We have scheduled this conference to pay a tribute to our distinguished colleague, Professor Inti Banerjee, whom we lost due to COVID. On her birthday, today we want to celebrate her achievements for research, which made difference for the society. As a medical doctor, nanotechnologist, and entrepreneur, she revolutionized the concept of nanomedicine by conceptualizing the products like the body oil for the massaging the children to deliver nutrients, iron and folic acid. Cosmetics like bindi, altas, sindoor to deliver nutrients for the women health. And the long-lasting contraceptive patches, tattoos for many, controlling the population, and likewise many other contributions which she made with over 40 patents to her credit. I must say and acknowledge that due to her prestige and reverence in the scientific fraternity, everyone to whom we invited for this conference, they happily accepted our invitation. And what we thought to have a small event actually turned out to be really a big event with 6 parallel tracks. And you can see all these distinguished colleagues from around the globe who are going to do deliberations today. As a convener of this particular conference, I thought to first walk you through the today's plan, what all sessions we have planned for you. So after this introductory session, which will be from 9 to 9:30, we are going to have 5 parallel tracks when we are going to have many distinguished scientists, faculty speaking to you about nanomedicine, drug delivery, biomaterials, proteomics, and digital health. We also have a very active student participation. I think we have seen over 800 or so registered participants yesterday night when I saw. And we have seen very enthusiastic response from the students to participate in the various competitions which we are going to organize today. So again, all these 5 parallel tracks will be running simultaneously, and students have been selected for the poster presentations, which they will present in the respective tracks. Additionally, if you have the proof of concept, product or pitch idea, there will be another competition which will be following the poster competition. And then we have various industry talk running parallel to different tracks. And finally, we'll all convene to the common session when we will have Professor Basu talking to us about information for society for biomaterials and artificial organs. After the lunch break, we have plans to do a hybrid event where faculty, staff, and students of IIT Bombay are going to meet at VMCC at IIT Bombay, while the online participants could still follow and join the event with the live streaming. Many of the faculty colleagues and students and staff are going to pay their tribute to Professor Inti. There will be various sessions about her lab research activities, and the various alumni and students working in her lab will also speak to us. And finally, we are going to plan to show two documentaries, in fact, one documentary and a short film. A documentary which conveys Professor Inti's scientific journey, which will be The Open Mind. And then we have a film on Professor Inti, her biography, which will be shown at the VMCC. Both of them will be screened today. So please do join for all these interesting sessions which we have made for you. And then in the evening, we have again 3 parallel tracks for the scientific sessions, which we will have many eminent speakers again interacting with you for various activities. Finally, we are going to have the valedictory session when we are also going to announce the winners of various competitions for today's poster and the pitch talks. We wish all the participants a highly stimulating event, and I'm sure that these scientific deliberations will ignite the young minds for the innovations. So good luck, everyone. And given the short of time, let me stop here. And let me now invite Professor Subhashish Chaudhary, Director IIT Bombay, who could not join personally, but he has shared his video, which I'm going to play for you.
Distinguished guests, speakers, my faculty colleagues, and students. On behalf of IIT Bombay and on my own personal behalf, I welcome you all to this special workshop, Translational Research in Medicine, which is being held in memory of one of our most distinguished and colleague and a friend, Professor Rinti Banerjee, whom we lost last year due to the pandemic. And it's today we come out of the mourning to celebrate the thing that she has done for IIT Bombay, the contributions that she has made to the society, to her students, to the society in general, as well as to the friends in our campus. She has been a very, very different personality. Someone coming from medical fraternity, taking up some kind of new initiative where she wanted to learn engineering. And for that, she chose IIT Bombay. And after that, she leveraged it. Her knowledge of both medical science as well as engineering to come up with a completely new researcher in our IIT Bombay system who would help not only our colleagues in biosciences and the engineering department, but in many other interdisciplinary programs where she would play a leadership role in defining what kind of research we should be doing, what would be the corresponding translational activity that we must indulge in, and also what would be the benefit that the society gets. It was a big loss for us, our friends, to the students, to the colleagues, and to me as a director, someone who was so precious to IIT Bombay to have lost someone like Professor Rinti to to this pandemic. We believe that IIT Bombay has gained tremendously from her role as a professor, as a researcher, as a mentor. And today, we have assembled here in this particular program to take a look at the mission that remained unfulfilled in her dream, where we thought that we could translate many of our research output in the form of translation that would benefit the society. We really mourn her. We really needed somebody like her to be with us for the journey of IIT Bombay. Unfortunately, that's not what it would be, and we have lost. But our journey must continue. The journey of all the scientists, researchers, our students, and faculty members at IIT Bombay to fulfill her dream, to see that the benefit of research of IIT Bombay reaches to the common people, to the society, and make an impact. That's what as IIT Bombay would always be wishful in its thinking. I hope that the deliberations that all of you are would be involving yourself today would be proved to be beneficial, and we look forward to this. My apologies that I am not able to come to this particular program in person because of some other preoccupation, but my mind will be always here with you as you discuss the way forward and how we can leverage each other's strength in making sure that our research in medical and other areas become more translation in days to come. Thank you so much, and I hope all of you will definitely benefit from today's program.
All right, now let me welcome Professor Suhas Joshi, Director IT Indore. Professor Joshi was earlier Dean Alumni Corporation at IIT Bombay and have seen Professor Inti's role very closely in different activities. So I invited him to share his experience and some of the thoughts for Professor Rinti Banerjee. Professor Joshi, if you are there, please join us and speak a little bit about Professor Rinti.
Me? I have joined. Hello?
Yes, Professor Joshi, yeah. I request you to now, say a few words for Professor Rinti.
Sure. So thank you, Professor Srivastava, for inviting me for this conference.
Spend years in laboratories testing a new treatment using animal models. This is sometimes referred to as T0. If the treatment shows promise, researchers will give the drug to humans for the first time. This step is
I think there is some
called T1.
Right. We request for all the participants to please mute themselves so that we can have the session uninterrupted. Professor Joshi,
yeah.
apologies, please start again.
Yeah. So thank you very much again for inviting me for this event. Professor Sanjeev Srivastava was at IIT Indore last week, and he proposed this idea that I should give at least a small talk about 4 to 5 minutes. And I readily agreed because I had a lot of respect for Professor Rinti Banerjee and her, her activities. In fact, I am not from biosciences or bioengineering. I'm from mechanical engineering. But I came to know about her when I became a Dean of Alumni and Corporate Relations at IIT Bombay in 2018. At that time, we were trying to, resurrect those CSR activities, and we are looking forward to some kind of proposals from faculty members. And in the first attempt, we got about 10, 15 proposals, and we sent those proposals to various public sector enterprises, and we found that Rinti's proposal was the one which actually caught everybody's attention. It was related to the on-the-site detection and care of waterborne diseases in a tribal areas, along with one of the NGOs, and that was like taken up by some of the public sector undertakings, saying that this is the proposal which is most relevant to us. The following year, we had more than 50 proposals, and again, Professor Inti Banerjee submitted some proposals related to this, and they have always caught attention of the companies and corporates. After that, we started, quite long interactions. I had long interactions with her, and I spoke to her. She came, and I tried to understand her work. And subsequently, after a while, the COVID started, and there was some lull in the activities. But at that time, we thought of an idea of what is called as Translational Research Academy for the institute, that is IIT Bombay. And at that time, we formed a committee, and I talked to Professor Chaudhary, saying that Professor Inti Banerjee should be there in this particular committee. And this committee actually called for the proposals from various faculty members, asking them for the translational research activities or the research activities which you are going to take forward in coming future. And we got about 50 entries from the professors, and we interviewed almost like 26 of them with the help of some eminent persons. And we found that Professor Rinti Banerjee was something which was completely different from, completely ahead of all the faculty members and their technology readiness level, from the point of view of technology readiness level. So her preparedness, her technologies, her projects were completely different, and they were close to the market, very close to the market. So when we started discussing this Translation Research Academy, she actually participated a lot in the developing the framework of this academy, and our job was to provide this framework and attract funding from the donors. So right now, this academy is being planned, and it requires a lot of funding, so it is taking a little bit of time, but I'm sure sooner or later this academy would take a shape in the institute, and we will appreciate a lot of efforts of Professor Rinti Banerjee. Then the third time we came to establishing this digital health center, Koita Center for Digital Health. Again, we were discussing with Professor Chaudhary, I was discussing with Professor Chaudhary how to form, what to do and all that. And then again, we zeroed upon Professor Rinti Banerjee, saying that she is the person who can lead us to the establishment of this Koita Center for Digital Health. And we discussed with her. She started participating with Professor Ganesh Ramkrishnan, Sivli Mukhopadhyay, and all other faculty members of the group. And she gelled so well with everybody. We had interactive workshops with Tata Memorial Hospitals, and she had taken a command of the whole center. And we started with her this center. Only the formal announcement of she being appointed as a center head was remaining. And I reminded Professor Chaudhary at some point that, Professor Chaudhary, why don't you send a communication to all the faculty and students that she is appointed as the head of the center. But suddenly at that point, we came to know that she is admitted to the hospital. And then we came to know that a few days later, we came to know that she is in ICU. Then Professor Chaudhary and we were, kind of a debating when person is in ICU, we will hold the announcement for a few more days once she comes out of ICU. And we did that. I mean, after that, she came out of the ICU, and the moment she spoke to Professor Chaudhary, we sent this communication saying that she is appointed as head of this Koita Center for Digital Health. And all the events turned, in a few days' time, all the events turned upside down, and we just lost her in this whole, matter of few days. We were expecting that she will come back and she will work for us, but yes, that was the reality, was something different. In fact, in all those discussions with Professor Chowdhury and deputy directors and all that, the institute was planning many things with the help of her. I mean, institute was so much dependent on her that we felt that she also would lead the IIT Bombay for making a the medical school. When as and when we ventured into getting a medical school in IIT Bombay, she should be the person who will lead us in that particular venture. So such was her, aura and capabilities, intensity of working, a wide variety of jobs she had taken in all these years. And whenever I requested her, saying that, Riddhi, can you take this project? I mean, she never said no to me. She never ever said no to me. Although she was busy, she was having many things at her hand, but she never said anything, no to me. Personally, I feel I'm at a great loss, and a very sincere, very honest friend, a colleague has been lost in the whole process. My heartfelt tributes and thanks to all her colleagues in BSB department, who are colleagues, and her students, who are actually making this conference of translational research in medicine TRIM. We were trying to do something TRA, Translational Research Academy in the institute. You are making a conference in TRIM. It's just something similar. I think she was a pioneer in the translational research. There is a valley between academia and outside world. We call that as the valley of death. And she has learned the way to cross that valley of death far earlier than many of the professors in the institute. So again, thank you for giving me the opportunity to talk on this forum, and share my memories with Rinti, and my best wishes to all the faculty and students who can for taking her work ahead through this conference. I'm sure you will have a great success. Wish you all the best. Thank you very much.
Thank you very much, Professor Joshi, for sharing your experience and these kind words. Uh, let's now move on, and let me invite Dr. Dennis Lee, the Senior Program Officer from the Bill and Melinda Gates Foundation. Professor Inti was awarded this very prestigious project from the Bill and Melinda Gates Foundation, and Dr. Lee has kindly accepted our invitation to join for this event. Dennis, if you are there, please say a few words.
Thank you, Sanjeeva. Let me start off by just sharing my screen. I just pulled together a couple of slides to share with everyone today. Okay. All right. So it's certainly a privilege for me to provide one of the opening talks to this conference, honoring the life and the works of Professor Rinti Banerjee. Rinti was what I would say an ideal grantee of the Gates Foundation. Her background as a physician researcher, her passion for solving difficult health problems for women and babies just made her a pleasure to work with. I was fortunate enough to be involved with this grant with Rinti for almost 3 years. And my reflections of our regular meetings with Rinti and her group is that she was always a very patient and kind individual who empowered her students during our meetings to provide scientific updates during our quarterly updates. And progress, as I'll show you in the following couple of slides, was really good. The work is ongoing. Sanjeeva was kind enough to take it on and continue some of the work towards the end of the grant. And there's some pretty exciting data that came out of this grant, which I'll share with you. So the grant, which was given in late 2018, was actually centered around the development of a 6-month microneedle contraceptive patch. This was really important for the foundation strategy in low and middle income countries. Some of you may know already that in the contraceptive space, the availability of long-acting products can address a lot of challenges in delivery of products to women who seek them. If you can come up with longer-acting products, you can address issues of adherence, alleviate the burden on healthcare systems. And what a 6-month microneedle patch offered was an opportunity to put a product in the hands of women who could then self-administer the product once every 6 months and lessen the burden of having to visit healthcare clinics on a regular basis. So the foundation actually put out a request for proposal at the beginning of 2018. And one of the nicest things about the grant that we gave out to Rinty was that we were not aware at the time the RFP was put out that Professor Banerjee was interested in working in this area. And at the end of the RFP, we gave out 5 grants to different organizations. The greatest surprise was the one that came from Rinty, because we were not aware of the work that was being done. It was an outstanding proposal, and we decided to fund it for a couple of years. So a little bit of background on microneedles. These are basically small patches, maybe ideally around the size of your thumb. And on it would be an array of tiny needles. And the design that Rinty's group went after was one in which the needles themselves, depicted here in yellow, with the patch on it, would be applied to the skin. The needles would detach from the patch, and then a long-acting formulation would remain in your intradermal space and release drug for approximately 6 months. The advantage of a product like this is that it could be self-administered. You get a long-acting duration. It's very discreet. Women can self-apply them. They're less painful than a regular standard injectable syringe device. And from a safety standpoint, because the needles themselves are the formulation, there's no potential for reuse of this device, and there's no sharps waste. But as I said, there were tremendous technical challenges associated with developing this product. And this was what Rinty and all our grantees had to manage during the grants. The first technical challenge, well, it's more of a challenge for the whole industry, is that there has yet to be a microarray patch or a microneedle approved for any use. No therapeutics, no vaccines. There are some coated microneedles that are in phase 3 now, delivering small molecular peptide drugs. Vaccines are in phase 1 and 2, but no dissolvable microneedles trying to deliver for an extended period of time. And so by going after a long-acting contraceptive microarray patch, the long-acting nature of it actually adds an additional level of complexity to the development of such a product. In addition to solving those 2 problems, one needs to figure out a way to apply them in an efficient and reliable fashion so that you get robust delivery of the drug. And eventually a development process, the development of a manufacturing process would need to be attained. And to top everything off, this would have to be done at a very low cost of goods. This is a figure of the type of patch design that Rinty's group designed, and shown here in a magnified figure are an array of microneedles. there's dye in it so that you can see the protrusions more easily. That was actually manufactured in the lab. So what was actually done? So in the proposal, this was the work plan that was set out. The work plan had two parallel tracks. One of them was the development of liposomal encapsulated drugs. The other one was the development of polycaprolactone encapsulated drugs. Ultimately, these two nanoparticles were combined into a mixture within a microneedle matrix to give the optimal profile desired for delivery of 6 months. Oops, let me go back. And on the right-hand side of this slide, I just show you a little bit of data that was generated from this work. This is cumulative release of drug from two optimized formulations. And you can see that there's beautiful linear release that attains about 100% release of drug at about the 6-month time point. This was tremendously exciting data, and this was attained probably as fast as any of our grantees from an in vitro standpoint. More recently, these formulations have gone into a pig study, completed the 6 months, and the samples are presently undergoing bioanalysis to see what the translation from in vitro to in vivo is. So very exciting progress, and probably within mid-year or a little bit later this year, we'll have a full idea of how this performs in the pig. And of course, ultimately, the goal is to get something that looks good in vivo so that we can eventually put it into IND-enabling studies to get it into the clinic. So let me just close off by saying and addressing Rinti's husband, Bhagwati, other family members who are maybe at the meeting today, friends and close colleagues. Thank you for allowing me to join the research community in celebrating Rinti's research contributions towards addressing important healthcare challenges for those that are most in need and honoring her life at this conference. Thank you very much.
Thank you very much, Dennis, for your kind words and giving an overview of very impressive work carried by Professor Rinti and her group. Let's now request Professor Ganesh Ramakrishnan, professor in charge of Koita Center for the Digital Health, KCDH at IIT Bombay, to say a few words about Professor Rinti and her association with KCDH. Professor Ganesh, over to you.
Professor Sanjeeva, can you see my screen? I just shared it.
Please share it now. Yes.
Thanks. At the outset, I would like to again thank the organizers of the RB Memorial Conference on Translational Research in Medicine as a very apt token of gratitude to Professor Rinti Banerjee, and specifically, I would like to highlight Professor Rinti Banerjee's contribution to the Koita Center for Digital Health right at its inception, as was very nicely summarized by Professor Suhas Joshi. I'll also talk about the shape Koita Center for Digital Health has taken up at IIT Bombay as a first of its kind center in India, more as a token of offering a gratitude to Professor Rinti. This focus on state-of-the-art and futuristic areas spanning healthcare applications, which could be predominantly software, EMR, EHR, medical imaging, etc. Healthcare data management, which is a lot about processes, standards, and the entire data management effort, which India as a large country is going to face. There's also the area of healthcare analytics, AI/ML, and algorithms. Consumer health and telemedicine, where Professor Rinti and several of our colleagues have had a lot of role to play. Computational biology and bioinformatics, and population health and public health policy. One of the key contributions of KCDH is that it partners with healthcare organizations, hospitals, healthcare providers, and also frontline workers in the form of ASHA workers and so on. I think Professor Rinti really brought in that strong perspective of hospital partnership and got the entire hospital partnership drive going. Fortunately, we've also been able to now work with the government at state and central level, largely through the Ayushman Bharat Digital Mission, ABDM. The areas of collaboration with digital health partners, which Professor Rinty helped chalk out in its early stages, span academic, research, policy, and funding. On the academic front, we've already got started with trying to design joint academic programs with Tata Memorial Hospital, for example, the joint MD PhD program. It's a mammoth task. Yet, I think Professor Rinty Banerjee made the initiation of this entire process a lot seamless. Needless to say, research is pivotal to the center, and research which is translation oriented, which also brings in data from the healthcare partners and delivers algorithms, delivers frameworks for analyzing data, including the most recent national disease database, towards which the Bill and Melinda Gates Foundation has also helped catalyze the process. We are very happy to announce that we have had engagement with very prominent digital health partners, some of them listed here. Several of them are hospitals, several of them are leading medical research organizations, companies. We're very grateful to Mr. Rizwan Koita, the founder of CTS Tech, who also made this center possible in the first place. Mr. Rizwan Koita and his wife Rekha Koita have also been pillars of the center and its functioning. And last but not the least, we've had excellent reception from the central and state governments through this Ayushman Bharat Digital Mission, where KCDH is playing the pivotal role of capacity building. Finally, we are very happy to have a diverse and global leadership in our advisory board. Several of them, thanks to Professor Rinty Banerjee's connections and long-term relations. We are very happy to have some of them even speak today out of gratitude for Professor Rinty Banerjee. And here's a track 5, the digital health, which is specifically being organized by our Koita Center for Digital Health. Professor Sudeep Gupta, Mr. Girish Raghavan from GE, Dr. C.S. Pramesh. Dr. Sudeep Gupta and C.S. Pramesh will also highlight the interactions they had with Professor Rinty Banerjee. And last but not the least, we'll also have talks in the evening with by Professor Brahman Mukherjee and Dr. Nitish Thakur, who again talk about some of the contributions of Professor Rinty Banerjee. We also have industry talks by Mr. Nipun, co-founder RIA, and Mr. Shashank, a founding member of Cure.ai. Between 11:50 and 12:10, we'll have poster competition by the students, both the IEEE and minor students, the kind of work they've been doing with the center since the last 1 year. Between 5 and 7:20 p.m., you'll get to hear from several faculty members associated with the Koita Center for Digital Health on the kind of research projects they've embarked upon, several of them funded by the Koita Center for Digital Health. They will also probably present some of their background work to get you excited. And we have students largely connected through our minor and IEEE programs today, which are fully running. Starting this year, we have the PhD program running. We'd like to invite a bright, excellent PhD applications here. And hopefully in the future, we'll have an MTech and MS program in health informatics floated. We are very grateful to our strong and diverse faculty leadership. The URL is presented here, and you'll get to see several of them today, either chairing sessions or presenting. Thanks. Again, my heartfelt gratitude to Professor Rinty Banerjee and the conference organizers. Thanks, Professor Sanjeeva. Over, back to you.
Thank you very much, Professor Ganesh. Let me now request Professor Rohit Srivastava, Head of Department of Biosciences and Bioengineering, to say a few words for Professor Rinty.
Yeah, thank you very much, Sanjeeva. Thank you for organizing this and managing it so well. I'm actually very, very sad today because, what Rinty did for the department, what she stood for, we have lost all of this. When I joined IIT Bombay in 2005, Rinty was our mentor. And she was a mentor to all the faculty who joined subsequently after me, especially in the biomedical engineering side. Our department is biosciences, bioengineering. But for the biomedical engineering side, Rinty was the pillar. She was the go-to person for any doubts that any one of us had and any proposals that we used to submit, we used to run it by Rinty because we knew that she had the insight that many people don't have even today. So just having that medical background plus getting a master's and PhD in this area, Rinty had tremendous knowledge and a wonderful, I'll say, connect with the medical fraternity. So that helped many of us grab really large grants, big, I'll say, connections, all due to Rinty. And most importantly, many of our earlier students, when I joined in 2005, many of my earlier students were co-guided by Rinty. Many of her students, most of her students, I was on the RPC, or the Research Progress Committee. So both of us knew what each one of us was doing. And being junior to her, we were really benefited by her presence. So this is a really, really big loss to the department. I don't know how we will ever be able to come out of this loss. And I hope that one day, her students in different parts of this country, in different parts of this world, will make all of us proud, because that's what Rinty stood for. She actually empowered her students, as was said earlier, and this is what we have all learned from her, and we keep doing it even today. So once again, let me welcome all of you to this conference, to this meeting, and to this memorial for the late Professor Rinty Banerjee, and a very big thank you to everyone who has agreed to give a talk at this conference. So thank you again, Sanjeeva, and thank you, everyone.
Thank you very much, Professor Rohit. And we are just slightly late for conducting the scientific sessions. I'm just pasting the various links to the participants. Now we are going to have 6 parallel tracks, 5 sessions, and for biomaterial there are 2 tracks. So nanomedicine track will continue with the same link, while the students and participants who want to join the other lectures can now slowly move on to the other team's parallel sessions. I'll just take a minute break, and then we will start this session by introducing Professor Rohit Shivasta, who will be the first speaker for this session. We also have 2 other eminent speakers lined up for this nanomedicine track, Dr. Kazunori Kataoka and Professor Dhirendra Khatti. So they will be speaking after Professor Rohit. So in 1 minute, let's do the shifting of different tracks, and then we will start with this particular nanomedicine track shortly. Let me thank all the speakers for the first session, all the distinguished colleagues who have shared their experience and their memories with Professor Rinty. Let me thank all of them for joining in today and conducting the session very much on time. We are now going to start the scientific deliberations. So thank you all once again for the morning session. We are now splitting into various tracks and starting the scientific deliberations shortly. Thank you.
Thank you. Bye.
Ladies and gentlemen, our next session is about to begin. May I request you to kindly mute your microphones and wait as we prepare for the session. Thank you. Ladies and gentlemen, our next session is about to begin. May I request you to kindly mute your microphones and wait as we prepare for the session. Thank you. Ladies and gentlemen, join me in welcoming Professor Rohit Srivastava from Indian Institute of Technology, Bombay. Professor Srivastava completed his PhD in biomedical engineering from Louisiana Tech University in the year 2005. Currently, He is the head of the Department of Biosciences and Bioengineering at IIT Bombay and also the Himanshu Patel Chair Professor at IIT Bombay. Dr. Srivastava is the proud recipient of the esteemed Shanti Swarup Bhatnagar Award in the year 2021 for Medical Sciences. He has received Bill and Melinda Gates Foundation GCE Phase 1 starting May 2013. Dr. Srivastava's lab have been placed third in the prestigious Healthcare Innovation World Cup organized by the HIT Lab on solutions to the management of diabetes with the cash award of $5,000. He is the recipient of the prestigious DBT IY by BA 2009 award and also been the elected member of the National Academy of Sciences in 2011. His research interests are medical diagnostic devices, nano-engineered materials, photothermal cancer therapy, and many more translational areas. Please welcome Professor Rohit Srivastava from Indian Institute of Technology, Bombay.
All right, Rohit, welcome once again, and now we are eager to listen to your scientific talk.
Thank you very much, Sanjeeva, and thank you again to the organizers. Once again, my heartfelt gratitude to you and the team for organizing this memorial conference for the late Professor Inti Banerjee. This is a very, very big step towards, her making her memory alive and keeping her memory alive in the, minds of all the faculty and students of this country. So once again, let me share my slide and I'll just get started. And I'll try to go fast so that we don't go too slow in the whole conference. So if you can see my slide.
Yes, please make it full screen. Yeah, you're good to go.
Okay. So if it's full screen now, let me again welcome all of you to this conference. My name is Rohit Srivastava. I'm currently heading the Department of Biosciences Bioengineering. And the talk I'm going to give today is affordable healthcare technologies. Just like Rinti, our lab also works on translational technologies, and we've learned a lot from Rinti. And this is, a very big honor for me to be speaking at her memorial meeting and to be showing what results we have got as part of translational work being done in our lab at IIT Bombay. So, just for the students and faculty, from different institutes who have joined in, let me give you a brief overview of the innovation ecosystem at IIT Bombay. It's important to understand that any institute like an IIT, be it IIT Bombay, IIT Delhi, IIT Kanpur, IIT Madras, I'm not taking names of all the IITs, but any of these IITs, you will have different departments in the institute. And these are departments which are either engineering departments or science departments. What you have to do is make sure that you work as a team and bring together faculty from different departments together. Only then translation is possible. Translation as a word is very simple, but as a concept or as an application is very difficult. You have to also bring in people from the translational side, which is, you have to bring in a business incubator, you have to bring in an entrepreneurship center, you have to bring in different funding agencies. You have to also rope in private funding partners, because if you want to really take your idea all the way from a proof of concept to the market, you have to have all these different, I'll say, blocks contributing to your idea. And then you have to have tie-ups with medical institutes. So this is where we feel the loss of Rinti a lot, because she was the medical go-to person for all of us, and she had answers that many of us don't, sitting as engineers in the department. So the medical institutes play a big role in translation of any research in this country. There are, of course, other engineering colleges that you can tie up with. There are incubators that you can tie up with. There are several that BIRAC has now funded across the country that have contributed to the innovation ecosystem of this country. And of course, once you have gone through a certain step, that is, you have seen that your proof of concept has been developed into a prototype and is now moving into academic clinical trials, and then is moving into actual trials at hospital under the CDSCO filing, then you have to make sure that you have tied up with either your own student startup company or transferred the technology off to a medtech industry. because this is where you have to cross the valley of death. If you don't do this at that point, then most of the ideas will just die at that point. They will never reach the market. So make sure that you have a company with you, you have a large company associated with the idea from the beginning, or you have somebody who wants to take that idea forward into the market. All the filing needs to be done from the company side. All the CDSCO filing has to be done from the company side, so that you get your test license, and then you get your manufacturing license to actually start manufacturing and putting it into the market. In this process, you will make many indigenous products, you will make affordable healthcare technologies for this country, and also create very high value jobs. So this is something that all of us should aspire for and learn from what Rinty has taught all of us. So just giving you a brief overview of healthcare challenges. If you look at the right hand top corner, you will see health expenditure per person. Look at Brazil, Russia, South Africa, and then look at India. We are at $75 per person, and this is in dollars, so you can convert that to rupees, but very little, maybe 5,000 rupees or something like that per person. And of that, 70% is private spending, only 30% is public spending, which means that anything that we work on in the lab, in our closed environment, has to finally match what the Indian population is going to spend. So you have to understand that all the probable solutions now, which comes up on the left hand corner, has to be affordable devices, has to be early, has to be simple reagents preparation, storage conditions, small turnaround time, and many things, because unless you make it affordable, it's not going to hit the market. And when I say affordability, we've got numbers now which actually say how affordable. So we've done many things in the past. These were our major research areas. We started working with magnetic chemohypothermia with Professor Bahadur's group, who was a mentor to all of us, when we joined IIT Bombay. And his group, our group, and Rinty's group did a lot of work together, had a lot of joint students, and published many papers and patents together. Then, we went into photothermal therapy for cancer, which is what I started off with Rinty's group. And I had a joint student with Rinty, Dr. Arvind Rengan, who's now at IIT Hyderabad and is doing very well. So all the help that we got from Rinty is now bearing fruition at different institutes, different places across this country and across the world, that you will now see in the form of her students doing well. There are many things that we started with her: drug delivery, nano and micro multifunctional materials. And then at some point, we started moving into my core research domain, which was diagnostics and devices. And we started working in affordable paper-based diagnostics, in microfluidics, in many biosensor areas, and so on and so forth. But then COVID hit, and this is where I think Rinty was at the forefront of all the research being done in the department. She actually was able to commercialize many and transfer many technologies from her lab. We also started doing many things during that time: IoT-based healthcare solutions, protective equipment, COVID care research, disinfectant sterilization products, critical care projects, and many more. And many of these products are now being tech transferred to industry. Many people are interested in technology licensing these products, and we are hoping to see many products in the market very soon. We have many technologies that we have developed over the years. And again, like I have always said, I owe many of this to the guidance that we got from the late Professor Rinty Banerjee, because she always put you on the right track. If you went to her with a medical problem, with a solution, she would always ask you the questions that a doctor would ask you, any clinician would ask you. And you had to have answers to those. If you did not, she used to give you the answers, and those answers actually made your life easier in trying to design these products. So we've done many products over the past. I'll go through them. This is just a snapshot, but we've started many startup companies from the lab, starting from Biosense to Neodox, which is most recent, to CareNX, to Norditech, to Patanjali, to Kavach, to Muzen, to Dynasense, and many more. We've now started companies with Arvind, with Professor Belare, and there are several more companies that have started up in the recent past. Some technologies that we were able to commercialize, and the biggest one is there in the center, which was the first device that came out of our lab called UCheck, and this happened way back in 2013. The project started in 2011 from a MTech student who came to my lab and said, "Sir, we want to do, we would like to do something translational and take a product from concept to market. And this was the IYBA grant that I got from DBT, very prestigious grant, but then it focused on translation. And that was a buzzword in those times. 2011, imagine, 11 years, 12 years back, people were just starting to look at translation. Me and Rinty were, of course, at the forefront, and Rinty was always leading from the front. So many things we started doing. I went into diagnostic side, she went into the nanomedicine side, and most of us, most of the efforts we had were focused on getting these products to the market. So the U-check is a big example. I'll show you that. The Sync is another big example, which is our own indigenous glucometer. And then, my students also did the Touch B, which is the hemoglobin monitoring system, which is using a non-invasive system. We also worked on a Wellcome Trust funded grant. As was mentioned, I got this HIT Lab funding from a HIT Lab award from New York. And this award actually gave us an opportunity to present before Wellcome Trust in London. And we got SmartSense. SmartSense is a full device, which can do point of care sodium, potassium, chloride, bicarbonate, pH, urea, glucose, creatinine. So this could be a perfect device for sitting in an ICU where the turnaround times actually matter a lot. So we also got an imprint grant to do Electrofinder, and then several other translational grants. I'll show you some of those. So the journey started, with U-check, as I said. But most importantly, please remember, whenever you're doing translation, always make sure that you survey the market. If you do not survey the market, you'll end up with a solution that nobody wants to buy. So you always have to find the problem and then find the solution. Do not start working on a solution first, because what idea for the solution you have in mind may not work with what the population actually wants. And the best way to do that is go to these path labs, go to these small hospitals, go to these clinics, go to these pharmacies, and find out what they want in that area. This is a big example. We actually surveyed labs. We found the problem. We actually found that there is a big problem with urine dipstick testing. And many of us just give samples today. We don't even understand what is happening there inside the path lab. But many of these small path labs sitting in small towns, not the large cities, but small towns, will always have problems because they cannot afford a urine analyzer. If they cannot afford one, how do they actually test one? So they actually get a strip and then they do a color comparison across the chart. This is always there in most of the strip boxes, but that's not the way to do it. If you're doing it that way, that means there is some problem with the way you are actually giving out data to the patient. So we had this opportunity in front of us. We started thinking about the solution, made a solution which looked like a small cell phone sitting on a box. And there was nothing to it, no innovation, when you look at it, but it was actually a lot of innovation. This was the first example of digital health. And this was the first example of how mobile phone could be used for diagnostics way back in 2010 and 11. And, we were able to commercialize it after due filing in the DCGI. And this came in the market in 2013. We made our own strips. We made the reader, and we started putting it into the market after the trials that Biosense Technologies conducted at KEM Hospital. We also went on to do a 10+2 parameter. The 10 parameter is the one you see on your left side screen. The 10+2 is the albumin to creatinine ratio, and we actually made these strips which were 1/3 the cost of what you can get in the market. So important thing is you can make anything you want, but don't put it in the price range that you will not be able to sell it in the market. You have to always price it correct. And this is something that was told to us at Wellcome Trust in London, that if you price it very cheap, then it will look like a Chinese product. If you price it very high, then people will just go ahead and buy the international product or whatever the competitor product is. Price it somewhere in the middle and it will always sell a lot. And that's what happened with these products. We were able to sell them a lot. The company made a lot of money from the U-check. Went on to do another project with ICMR where we picked up a challenge of glucometer. And this is a big old challenge. Even today, strips are available at 18 and 30 rupees per strip. People cannot afford them. If they have severe diabetes and they have on insulin, they have to actually test themselves 5 to 7 times a day. The solution, you have to make strips which are affordable. You have to make a reader which you can give away for free. And that was what we started off with. You have to also look up what exists in the literature. Please do not work in a patented area, because if you do that, then some company is going to file a patent and your product is always going to be in trouble. It will never see the day of the light of the day. The fact is, if you can look up the patents, and this was a simple one because the patent that you see for this one is off patented, a long time ago. It's a glucose oxidase peroxidase method. There's nothing different to it. There are two dyes that are going to be used, and if you can use these dyes on the strip and be able to detect the glucose, then nothing like it. So we were able to make our reader, we were able to make the strips for it, and we were able to do the validation of it at different hospitals in India. And most importantly, the Clark's error grid that you see here, you have to show data that your device versus the reference device that you're going to use, more than 95% of that lies in the zone A. If you can do that, then you get an approval from it. You can actually put it in, you start packaging it, putting it in boxes and start selling it, in the market. Most importantly, again, this was cloud connected device. So anywhere anybody would be taking a reading, it would go on a cloud. Of course, anonymous data, but we would know which areas, what places, we're using this. And it was released in a very big function in New Delhi by the then Congress government, and ICMR made a big splash, got a lot of competitors who wanted to buy off the technology. We did not, and went ahead and made, newer versions for that. Then got the Wellcome Trust grant from London, where we were asked to make this device and affordable assays. The assays we made in cuvettes, because that was the best way of selling these assays. ICUs will always draw blood. There will never be a problem of drawing blood, but then you have to spin down the blood, because if you use whole blood, then all your readings go for a toss. So the sodium, the potassium, the chloride, the glucose, the assays are simple. It's all there in the literature. You're not using somebody's patent. It's all in the textbooks. So we are using the same assays, putting it in cuvettes, and just letting them and capping it. And then as soon as you get your blood, you spin it down, take the plasma off, and put it into this. Or whatever, the best way to do that is, you have all these reagents and everything available at the bedside. If you can do that at the bedside, no better thing, because you have your data, your results within 5 minutes of taking blood, which will actually save a patient's life. So this was held in very high regard by Wellcome Trust. And the device, the assays were made by us, the design was made and transferred to Wellcome Trust, transferred to the company to take it forward. We worked on several other technologies. I'm just giving you glimpses because I don't have time to give you all the detail. But we got a DST funded grant for kidney related disorders. This was for albumin and creatinine. I showed you one of the strips earlier, but those strips were with a mobile phone reader. This one, we are making another strip, different assay, but with a electronic reader. And we were able to make this. We were able to do all the testing of this at 2 different hospitals in the north, at PGI and AIMS. And we were able to show the data, this data to DST as well as to Grand Challenges Canada, for which we got another grant, where we showed mapping of the population in that area. We also worked on microneedle technologies. In fact, we got the Bill and Melinda Gates Foundation grant in 2013 for making a TB dot microneedle array. And Arvind started this project, and we actually completed this project successfully and gave all the results to IKP, which was handling the GCE phase 1 scheme from Hyderabad. And we started working on many other things. We started working on a vaccine delivery system using dissolvable microneedles, as Rinty's group has been also doing. And we have got terrific results with our dissolvable microneedles. There is at least 2 companies which are interested in licensing this technology. We also worked on silicon microneedles, not just dissolvable, but silicon microneedles, which are hard microneedles. We made them using a process at IIT Bombay as well as at ISC Bangalore. The needles look like the ones that you see on the right-hand corner, and they are actually as small as a 34 gauge needle, and as with a similar bore. And the most important thing is that they have the capability of piercing the skin with very little force. So once you can do that, then our idea was to combine these microneedles patches into a smart device. And we made a small smart device version for this and proposed an idea for a PhD student who started working on delivering drug across the skin using a smart device integrated microneedle patch. And we actually worked on that technology. We made different versions of that technology with micro pumps as well. This was the larger version. And you know, as you can see on the right-hand corner of your screen, what you can see is the profile that we could actually deliver drug at the touch of a button. So all you had to do was program the profile, and the device would do the needful. And you can have a reservoir, you can have a small bottle, you can have whatever in it, which can deliver as much drug as you wanted to using the microneedle patch. The final version looked like this, which is a more compact version, and then there are now people who are interested in taking this forward. We've worked on many technologies. This is another one, which is a big one, which is supposed to hit the market this year. And this is called CallCheck, which is affordable point of care solution for total cholesterol, HDL, LDL, and triglycerides. This was supported by PACE BIRAC. BIRAC has two schemes, a small 6-month, 18-month scheme, which is for 50 lakh, and then you come back to BIRAC with enough data that you can actually take it to the market from TRL4 to TRL9, which is called the CRS scheme, the contract research scheme, wherein the company will take the onus of commercialization of this device. So we made these devices, you know, we worked on the technology, put it on strips, put it in solution phase, made the paper-based assays, tested it out, made the readers for it, did industrial design for the readers, made the design for the strips and the readers, actually tested it out with hospital samples, as you can see on this slide, got excellent results, less than 10% variation. And if that is the difference that we are getting with our device, the liquid assay reader and this, you know, we got go ahead to take it into the next phase with a CRS grant from BIRAC. And at this stage, we are with a contract manufactured device, the data and everything is now starting off for trials at a hospital. So we hope to get this approved within the year end or maybe middle of next year so that we can put it in the market. We're also working on a cardiac troponin INT for ICMR project, where we are using lateral flow assays to do that. Now, many of you are aware of lateral flow assays. You can see 2 lines in them if something is positive. And many of you have seen that during COVID times. But can you actually read these lines? Yes, you can. If you make a reader around it, we actually made a cell phone-based reader which can read the intensity of these lines and give you values of the readings to the user. If you can do that and compare it to a comparator product, then nothing like it. So the trials are happening at a hospital, and hopefully we'll have data to show to ICMR very soon. We also made a hardware-based urine analyzer, which is now being tested in the lab, and hopefully will be released by the company. And this is my student's company, which is going to take it forward. Many things happening. We got a WRCB funded project for doing a diagnostic medical device for detection of glycated albumin. This is a big marker. If you look at diabetes, you've got a spot glucose, you've got a 3-month marker, which is HbA1c, and then you've got a 3-week marker, which is glycated albumin. If you can actually combine all 3 of them and make a glycemic panel, then you cannot believe the market that this will have in this country. So we've got the device, we've got the assay ready, and we're now hoping to actually make them into one device. Many technologies from the lab, as you can see, many of them are under clinical validation. We're working on several technologies with lateral flow assays. We're working on many nano-based technologies, which I'm going to show you in the next slide. So I'm going to just take a couple of minutes from now, just give you a brief overview of some of the nano technologies. Because this is nanomedicine track, I thought I should actually have this as well. All the stuff that I showed you was for faster reach to the market, but then there are many of these that take longer time to reach market, and we have not left them. We are making sure that many of these we are going ahead. We are setting up a GMP facility in the department with help from a donor who's helping us set this up through his donation, and Professor Joshi was responsible for getting the donor to the department. Many things are happening. If we are able to scale up the development, scale up the manufacturing of these different nano medicine products in a small GMP facility in the department, then all of this can be put into clinical trials. And then we will not need to actually have a company at an earlier stage. We can actually have a company at a later stage. So we are setting up a company around our gold-coated nanoparticles for photothermal therapy, and this is with Arvind's group and Abhijit Dey's group at Actrec. We are also doing a nano-engineered biodegradable bone and tissue fixation devices. We've already set up a company with Professor Belare and Kunal from chemical engineering. We've also setting up, we've transferred technologies. So initially we were trying to set up a startup around it, but both these technologies that you see at the bottom, the nano-engineered sponges, and the on-demand drug delivery system. We've actually transferred these technologies to an industry, and we are getting money to take this forward through development in our lab and then clinical trials through the industry. So there's a lot of emphasis. We are working on at least 35, 40 other technologies in the lab. I cannot even show you all of that. But if I actually had chance to show many of this, I will do that next time. Whenever this conference is held next year, I'll come back with more and more data. So what I'll do is I'll just skip some of these slides because I'm not going to show you. Are you able to see my slides still or not? Or did I escape?
Rohit, just now, I think you stopped sharing.
Sorry.
Until now, we were seeing.
Yeah, just go back because I want to actually just show. Sorry. Yeah. Is it visible now? So I'll just skip some of these slides. This is all data from all the nanomedicine work that our lab is doing. Many of it started off in collaboration with Rinty Banerjee's group, and many of it we are now continuing with other collaborators across the world, where we are getting terrific data, and we are hoping that many of these technologies can reach the market. So you see many things happening in our lab. Most of this has a translational aspect to it. And we always have a clinician associated with many projects that we do. We never ever do it alone. We always do it in a team with many other faculty from departments, so that we can take help from different departments, science departments and engineering departments. All of this you will start seeing now in some time, getting tech transferred from our lab. And we also do a lot of social work from our lab. We look at malnutrition, take many of our technologies that we've developed in our lab to do screening of malnourished children and maternal health screening at different camps in India and different places in India, in Maharashtra as well as northeast corners of this city. So we're doing many things with our student companies. The Care Mother Kit that you see in this slide is from a student's company, and many things that we've developed with our students' company. We're actually using that to do malnutrition screening as well as maternal health screening in different parts of this country. So once again, like I have been showing you device pilots in Maharashtra and Rajasthan. We've got data, we've got a lot of interest, we've got actually a lot of things happening with all our devices. We've got many grants funded to our labs, starting off with Bill and Melinda Gates, with BIRAC, with Imprint, with GYTI. Every year, many of my students apply for GYTI. This year itself, we got two of the most prestigious GYTI awards from BIRAC, and Stanford MedTech Award, Samsung Innovation, PACE BIRAC, HIT Labs. We make sure that we keep training students. So till date, I have graduated 35 PhD students. We have another 22 ongoing with me. Many of them came from Rinty Banerjee's lab, and I am very happy to be able to guide them. And many students are continuing their work in our lab through funded projects. Many staff research interns are being trained. Many innovators are being supported. And most importantly, we believe in filing patents, because all this technology means a lot to the company that takes it forward. So we always have to have a patent around it. We filed more than 150 patents till date. 25 of them have been granted, both US and Indian, and many more are on the verge of being granted. Many products have been commercialized, more than 20 such products, and many more we are working on, more than 40 products we are working on. Many startups we have created, and many jobs have been facilitated. We've actually been recognized at several places by the Honorable Prime Minister, by the Honorable HRD Minister, by the Honorable Science and Technology Minister, and at different forums in this country. We've done a lot of outreach activities. We've made sure that people are aware of the translational research happening in the department, and we hope that the legacy that Rinty Banerjee has left behind is always taken forward, from the department. Once again, let me thank all the PSB partner network and all the hospitals, all the IITs that we work with, the venture centers that we work with, the companies that we work with, both large and small, and my group, and most importantly, my mentors, Professor Padmanabhan, who's always been the biggest mentor to all the youngsters in this country, Dr. Mrs. Manju Sharma, who makes sure to mentor the youngsters so that they are always on the right path and always doing well for this country and always working for this country. And Dr. Renu Swarup, who did a lot for this country and for myself, during the last 15 years that I have been associated with BIRAC. Let me once again thank all faculty members, collaborators, students, startups, and once again thank you to Sanjeeva for inviting me to this conference. And a big gratitude to Rinti Banerjee for all the help that she has given to many of us when we joined IIT Bombay. Thank you.
Thank you, Rohit, for the wonderful presentation. Now my team will take over for the Q&A session and the discussions.
Thank you, sir, for so very informative presentation. I'm Lisha. I'm the moderator for this team, and with me I have Ankit, and we both will moderate this meeting. And we request you all, if you have any questions regarding the presentation, you can come up. We can read out your question. You can write in text, and we can read out the question to Professor Rohit Srivastava.
Rohit, all the participants are, of course, appreciating your work, and there are some comments I see that, okay, very impressive research for diagnostic products. So, of course, you have really taken Professor Rinti's legacy for translational research forward, and I'm glad that many of the students from her group are also continuing, and you're nurturing their work further and providing the mentorship to the translational research what they are carrying out. With this, I think let me thank you, Rohit, for joining us today for the introductory session as well as your research talk. And given the short time we have, I think we'll just move on to the next lecture now.
Sure.
Thank you, Rohit, once again.
Thank you very much, Sanjeeva. Thank you.
Participants, if you have any question, please drop us a message. We will convey to Professor Rohit, and I'm sure he will revert back to you. Thank you, Rohit, once again. Bye.
Sir, actually, we have a question in chat. Can I read out to you, please? Professor Rohit, can I read out your question?
Yeah, please do.
So one question in the chat is, how are you looking at the future medtech setup for in IIT Bombay?
How are you looking at the future medtech setup? Startup. So yes, see, startups are something that are several. Sign has been involved in many in facilitating many startups from IIT Bombay, and I'm also chairing the biotechnology ignition grant diagnostic panel for BIRAC for several years now, and we see a lot of people come in with individual ideas. We see a lot of startups come in with ideas that we would like to take forward. Many of them I am facilitating Sign to take forward. Many of them are being taken forward by other IITs. But I am very confident that in a few years' time, we'll see a boom. And even maybe starting this year itself, we'll see a boom in the number of healthcare startups that IIT Bombay will see.
Okay, so we will be starting our next. Yeah, so we will be going with for our next speaker.
So, may I start my lecture?
Yes, we will tell after the introductory session.
So, may I start my lecture?
Good morning. We are just giving a short introduction for you.
Okay, thank you.
I'll share.
Ladies and gentlemen, today we have with us Professor Kazunori Kataoka, who joins us from Kawasaki Institute of Industrial Promotion, Japan. Professor Kazunori Kataoka received his PhD in polymer chemistry from the University of Tokyo. Professor Kataoka is the Senior Vice President and Director General of Innovation Center of Nanomedicine, Kawasaki Institute of Industry Promotion. He was visiting professor at University of Paris, France, Tohoku University, Sendai, Japan, and Ludwig Maximilian University, Munich, Germany. He has been appointed as adjunct professor at Eshelman School of Pharmacy, University of North Carolina, Chapel Hill since 2015. Professor Kataoka's research focuses on developing smart, intelligent nanocarriers for delivering therapeutic biomolecules, with more focus towards drug and gene delivery systems by utilizing the concepts of polymer chemistry. Ladies and gentlemen, please join me in welcoming Professor Kazunori Kataoka.
All right, Professor Kataoka, welcome to the conference. Please share your presentation and start.
Okay, I will share my presentation. Please wait for a while.
क्या चीज? नहीं प्रीतम आ गया. हाँ.
Can you see my slide?
Not yet.
Not yet? Okay. Let's see.
Something is coming on the screen. Yeah, now we can. Yeah, now we can.
Can you see my slide?
Sir, it was visible sometime earlier, but not again.
Can you see my slide?
Yes, it has started, yes.
Can you see?
Yes, sir. You can see your screen.
I'm not sure. I just want to know whether my slide is sharing or not. Mr. Chairman.
Yes, sir, your screen is visible. Yes, screen is visible.
Something wrong. Can you see my slide?
Yes, yes, sir. It's visible. Yes.
Okay. So can I start my presentation?
Yes, sir, please.
Okay. Thank you very much. I hope you can hear my voice. So first of all, I would like to express my sincere condolences on the passing of Professor Rinti Banerjee, one of the very brilliant scientists in the field of nanomedicine. So it is my great honor to have opportunity to participate in this international conference for the tribute of Professor Banerjee. So first, in this lecture, I would like to talk about engineered nanosystems and nanoconjugates with smart functionalities for targeted therapy of intractable diseases. So our most favorite nanosystem is block copolymer micelles, as schematically shown in this slide. So when you dispose amphiphilic block copolymer in water, they self-assemble into core shell structured particles. That is a block copolymer micelles, and core segregated from outer environment work as a nano reservoir of drugs, which is totally surrounded by hydrophilic outer shell composed of flexible tethered polymer strands. Most typical polymer used for this purpose is fluorescent glycol. So in this way, you can make this particle have a stealth property under blood circulation. And you can manage the size in a size which is comparable to the size of a virus, which is a very big advantage for expecting high extravasating ability. So here is the TEM of the example of polymeric micelles we prepared. So you can see each micelle has a very narrowly distributed, and size is around, in this case, maybe 30, 40 nanometers. And of course, you can conjugate some ligand in the periphery of tethered chain to make this system have active targeting properties. And the chemical structure of core is also important. And for core structure, we usually utilize polyamino acids because polyamino acid is biocompatible and biodegradable. And just by changing the structure of side chain, you can manage the chemical properties of the core depending on what kind of cargo you want to deliver. So first, I would like to just visually show you how this stealth property works for longevity of blood circulation. So here we are now observing the thin capillary of earlobe of mouse. So we, the labeled platelet with green fluorescence dye and nanosystem, which is size lower than 100 nanometers. And if we inject nanosystem without PG, what we observe is like this. So you can see the red spots, which is size is comparable to platelets. Why this happen? It's due to the instantaneous aggregation occurs just after injection of this particle. So this is a very dangerous situation which may induce the stroke or embolization. However, if you totally cover this nanosystem with dense layer of PG, situation is totally changed as shown in the right side here. Now you do not observe any aggregates, and nanosystems stably circulate in blood compartments. So eventually, here is one example, the polymeric micelle we prepared. This micelle loaded with platinum-based anti-cancer agents. So compared to free drug, this micelle formulation of platinum-based anti-cancer drug have a longevity in blood circulation and half-life prolonged to maybe more than 12 hours. And this is very important. Here is the relationship of blood half-life of intravenously injected nanosystem and injected nanosystems returning the heart in one cardiac cycle. So 12-hour, the half-hour means 99.9% return to heart. That means only one single particle out of 1,000 particles captured under the blood circulation for the one cardiac cycle. And if you make this to 99.99, then you can achieve half-life of 1 week. But anyway, by prolonging the half-life, you can have a significant increase in the accumulation to solid tumor as shown in this right side figure, 20-fold increase. Why this happen? So this is well-known, the phenomena named enhanced permeability and retention effect. So compared to healthy tissue, the capillary in solid tumor has a very leaky structures, many openings. So that means if you make your system to have a stealth properties, then this stealth nanocarrier like polymeric micelle has opportunity to constantaneously accumulate into solid tumor with avoiding to distribute to healthy tissue, which may induce non-specific side effects. And actually, EPR effect works not only for nanoparticle, but also works for the antibody drugs. Everybody knows the now antibody drugs are utilized for the treatment of cancer, and also antibody drugs is a size around 20 nanometers, 15 nanometers, also accumulate by this EPR effect. So the effect looks very simple. However, what we should concern is that not only accumulation, we have to think about effective penetration into tumor mass. Now, many nanomedicine, including those developed by ourselves, are now have been in clinical trials. And then we encounter some difficulties. One of the problem is a heterogeneity in EPR effect. The EPR effect may be changed by the stage of cancer and the individual difference of patients, and also the the different type of cancer. And often the case in a clinical situation, there's a very thick stroma which works as a penetration barrier, which so this is the pathological section of human pancreatic cancer that is very, very heterogeneous. So this dark blue region, that is a cancer cell nest, which is totally surrounded by thick stroma colored red. And vessel locates only in the stroma. There's no vessel in cancer cell nest. So that means that even if your system extravasate, still the nanosystem needs to penetrate through this thick stroma to reach the final target. So how to overcome these problems? There may be three approaches. Number one is the size modulation of nanosystems. Second is the use of reagents enhancing tumor permeability. And third, use of ligands inducing active transport, that is a transport other than EPR effect. So I would like to briefly mention how size modulation of nanosystem works to improve the accumulation and penetration. So for this purpose, we would like to directly observe inside of the tumor. So we are utilizing in vivo confocal microscopy. So in this way, you can directly observe tumor inoculated in living animals. So after injecting fluorescence-labeled polymeric micelle by tail vein, we can observe the extravasation and penetration of polymer micelle into cancers. So here's one example of GFP expressing cancer with size around 3 to 4 millimeters. And green spots are corresponding to the nucleus of each cancer cells, and yellow vessels, that is a capillary. And you can now, in this way, you can directly observe the living tumor. So we are now observing the heterogeneous pancreatic cancer. So please follow the red arrow. This is a high-speed movie. So you see some random opening and extravasation, penetration of fluorescence polymeric micelles in solid tumor pancreatic cancer. So maybe you cannot clearly observe. So I will show you in the next slide the high magnification movie. So here's a high magnification movie. So left side is 30 nanometer micelle, right side is 70 nanometer micelle. Now you can see the difference. The 70 nanometer micelle, after extravasation, the color, fluorescence stuck in the vicinity of vasculature. However, 30 nanometer micelle, you see that this, diffusion of fluorescence occurs after extravasation. So here is a quantitative results. So in a 70 nanometer nanoparticle at a non-eruption site, there's no increase in the fluorescence. And in the eruption site, you see significant increase in fluorescence, which corresponding to the extravasation, but fluorescence stays there over time. However, 30 nanometer particle, after eruption, there's an increase in the fluorescence, but it diffuses out. And also importantly, even in a non-eruption site, there is a gradual accumulation occurs. So that means it's corresponding to the significant distribution for 30 nanometer particles. So this means there may be the dynamic vent as shown in this slide, and also static smaller pores. So, but large particle cannot, pass through the static pore. Only chance is dynamic vent, but it's stuck in the vicinity. But if you downsizing the micelle to 30 nanometer, it can not only accumulate, but also can penetrate into tumor. So that means that a small nanoparticle diffuse better than large particles. For example, this is a 100 nanometer liposome loaded with doxorubicin, red fluorescence. So in this case, you see all fluorescence stuck in the vicinity of vasculatures. And actually, here is anti-tumor efficacy. The 30 nanometer micelle only show the significant efficacy to suppress the growth of tumor. So here is some clinical development of micellar nanomedicines. The example which I just show you, cisplatin micelle, and another one, pyrolusin micelle loaded with anti-cancer drug, is in phase 1 to phase 2 clinical trial, and also the other system loaded with siRNA antisense oligo is in phase 1, and most newest one, messenger RNA system, is now in preclinical treatment. So in this case of cisplatin micelle, we are now doing clinical trial in combination with immune checkpoint inhibitor. What is immune checkpoint blockade therapy? The checkpoint proteins such as PD-L1 on tumor cells and PD-1 T cells help keep immune response in check. So PD-L1 binds to PD-1 and inhibits T cell killing of tumor cells. So by blocking PD-L1 or PD-1 by utilizing antibody allows T cell killing of tumor cells. So that is the ICB therapy. So it really works for treatment of many intractable cancer. However, still limitation. The response rate remains low in various cancer types, including glioblastoma and pancreatic cancer, and immune-related adverse effects. So the reason for the low response is tumor targeting ability is low due to the biological barriers, and some tumors are immunosuppressive, so-called cold tumor microenvironment. And also, off-target binding with healthy tissue is a big problem. So how to overcome these problems by nanomedicine approach? There may be ligand installation and nanocarrier encapsulation, and also by combination with ICD, that is immunogenic cell death, inducing drugs to turn cold tumor microenvironment into hot tumor. And as for the off-target binding treatment, we can make only the release ICB drugs only in the tumor microenvironment, but not in healthy tissues. So we are now challenging to treat the most intractable cancer, glioblastoma multiforme. GBM is the most deadly form of human cancer, resistant to immune checkpoint blockade immunotherapy, and mean survival is only less than just 1 year. And only 5% of patients survive 5 years. It's really in a very miserable situation. So first, I will show you how to change the cold tumor, immunosuppressive tumor, to hot tumor by utilizing immunogenic cell death. So what is immunogenic cell death? So some type of cancer drugs, kill cancer cells. And during this, dying process of cancer cell, the cancer cells secrete compounds so-called, dump. That is, the including calreticulin, HMGB1, and ATP. And this dump, stimulate the dendritic cell to turn immature dendritic cell to mature dendritic cell to recruit and the cytotoxic T cells. So as shown in here, the anthracycline anti-cancer agents like epirubicin, doxorubicin are very strong inducers of the ICTs. Fortunately, we already developed the doxorubicin or epirubicin loaded pH-sensitive polymeric micelles as shown here. So in this case, we conjugate the epirubicin by acid labile linkage to glucopolymer of PEG and polyaspartic acids by pH sensitive linker. And micelle is stable under blood circulation, but it liberates the active drugs at the acidic component of cancer. So it's already in a phase 2 clinical trial for the treatment of angiosarcoma, and we utilize this polymeric micelle in combination with ICB drugs. So we treat the P10 negative GBM. What is P10? P10 acts as a tumor suppressor gene through the action of its phosphatase protein product. A mutation of this gene are a step in the development of many cancers, including glioblastoma. So as shown in this figure, almost 40% of human patients, clinical patient, has a P10 mutation. So treatment of P10 mutated glioblastoma is very important. So we prepare the orthotopic GBM model of P10 negative cancer, and and carried out the treatment. And PBS doesn't work, anti-PD-1 very poor, and PDC micelle is somewhat better, however, still not so effective. But if we combine the PDC micelle and anti-PD-1, almost 90% of mice, survive more than 70 days. And by the MRI, no tumor detection at day 27 for mice treated with this combination therapy. So that is very, very effective. So second, the example is, improve the tumor targeting ability by ligand installation. In this case, we utilize glucose ligand to target glucose transporter 1 overexpressed in glioblastoma capillaries. And also, we make system to circumvent the immune-related adverse effect by TMA activatable release by utilizing disulfide linkage as a reduction signal in tumor microenvironment. So in this case, this is not micelle. We make a smart bioconjugate of anti-PD-L1. So as shown here, we conjugate anti-PD-L1 with polyethylene glycol, but some of the PEG chain has a glucose at the chain end, and linker is disulfide groups. So the strategy is following. So glucose conjugated antibody can, effectively accumulate into glioblastoma by a glucose transporter. And in the reductive environment of glioblastoma, the PEG liberates from anti-PD-1 by this, scheme, and recovery of antigen binding ability of antibody. And also, at the healthy tissues, the pegylated antibody doesn't bind to healthy cells and circumvent non-specific, side effects. So how it works? So first, I will show you how the accumulation. So here, the X stands for the percentage of glucose conjugated PEG in whole pegylating fraction. So 25% of glucose into whole PEG fraction shows the highest accumulation to tumor compared to 20 times increase compared to native anti-PD-L1. So another issue is how this selectivity. If antibody also accumulate into normal brain, normal site of brain, it may induce some side effects. However, as you can see here, this is the AUC ratio of tumor normal brain ratio. And you can see compared to native anti-PD-L1, the glucose conjugated and smart antibody shows more than 30 times selective accumulation at tumor site. So here's the efficacy. So saline, native anti-PD-L1, the PD-L1 conjugated with PEG without glucose doesn't work. And this one here, that is the conjugated with glucosylated PEG, but non-activatable CC bond doesn't work. And only the conjugate with smart, the smart responsivity shows a significant efficacy to suppress the growth of this very aggressive P10 negative, the glioblastoma. So eventually, 60% of mice attained tumor-free survival for over 90 days just by a single injection of smart bioconjugate with 1/10 of clinical dose approved for native anti-PD-1. And another important point is that this system also acquires the memory effect. That means the system works just like a vaccine and prevent the recurrence of glioblastoma. So in this experiment, first we inoculate the glioblastoma cell into right side of brain and treat it by the smart bioconjugate, and after brain tumor disappear, we quantify memory T cells and inoculate again with glioblastoma cells in the left side of brain and follow the survival. So here is a quantification of memory T cells. Compared to native mice treated with this smart bioconjugate shows a significant increase in the percentage of effector memory T cells. And after inoculation, re-challenge, of course, the native mice shows a significant, the growth of glioblastoma, but mice pre-treated with this smart bioconjugate doesn't show any progression of glioblastoma. And we had 80% of treated mice re-challenge the tumor. So that means it works, for the prevention of recurrence of glioblastoma. So how is the immune-related adverse events? That is a big issue. So almost 40% of patients show this adverse effects because of the, the antibody, the bind to healthy cells to stimulate T cell and macrophage. So this is a tissue infiltrated, the inflammatory lymphocytes, and native anti-PD-1, there is some significant increase in percentage, but there's no increase for the mice treated with glucosylated smart anti-PD. And this is a production of pro-inflammation cytokines, TNF-alpha, IL-6, IL-1 beta. It's a significant increase for the native anti-PD-1, but not for the, the smart bioconjugates. So in summary, by utilizing nanomedicine for the, epilevicin micelle as ICD inducer to change cold tumor to hot tumor, and also utilizing glucose as a ligand to crossing the biological barriers of blood-brain tumor barrier, and also circumvent the off-target binding by selective reduction at tumor microenvironment. So then I will, switch the gear to how nanomedicine works for the delivery of oligonucleotides, nucleic acid-based drugs. So this is a history of drug development. So small molecule drugs, the first synthesis of urea to first synthesis of drug takes almost more than 70 years. And antibody drugs, discovery of monoclonal antibody to first antibody drug in the market, it also takes 20 years. Always it takes time. And then here comes the nucleic acids, and it also takes 20 years. But nucleic acid drugs has a very, the promising because it can target also non-coding region of human genome. However, problem is in vivo stability and impaired biodistribution. They are critical issues for nucleic acid-based drugs. So again, we utilize this, nanomedicine for delivery of siRNA and antisense oligo to glioblastoma multiple by size modulation. So compared to the cancer which expect high accumulation due to E pyramid, always glioblastoma has a limited accumulation due to blood-brain tumor barrier. But even in this case, there is some chance for the downsizing nanocarrier system to snake into the parenchyma of glioblastoma. So we use this strategy. So compared to the other nanoparticle and liposome, one of the advantage of polymer micelle is that you can downsizing this, but still it's 14 nanometers. And of course, siRNA is very small, but due to this small size and so it's a poor longevity in blood circulation. So we realize this range, target range, size of antibody is very promising. It's around 20 nanometers. And also, if you make system to have a smart translation to clinics, all formulation process should be simple. Sometimes chemist makes a very, the complicated nanoparticle, but it's okay for writing paper, but not for the translation. So what we developed here is we call unit polyion complex, unit PIC. What is unit PIC? So when you mixing siRNA with oppositely charged block polymer, it spontaneously forms polyion complex micelle. However, this process is not single step. It's always two steps. First, by mixing siRNA or antisense oligo with oppositely charged block polymer, what we observe is the formation of the complex with smallest association number to compensate charge. That is a unit PIC, but unit PIC usually proceeds into the secondary association to form micelles. But we focus on the size of unit PIC. That is a very good size. So, but we need to prevent, stop secondary association, and also we need to improve the stealthiness to escape from scavengers in blood compartment of unit PIC. So our answer is here, very simple. We changed the PEG from single-armed PEG to two-armed PEG, and we regulate the number of positive charge of block polymer just as half the number of siRNA. And in this way, just mixing, it spontaneously form, this tetrahedral formed into PIC. Size is comparable to antibody. So you may wonder, such a simple system really works under blood circulation. So I will show you the results. Now we are observing the thin capillary of experimental animals under in vivo confocal. So we utilize the fluorescence resonance energy transfer. So we conjugate the donor and acceptor pair of fluorescence dye to both ends of siRNA. Because of the distance is 5 nanometer, so if you excite the donor, the energy transfer to acceptor, and you observe the red color. But if siRNA degraded, there should be no red, so what you observe is just blue color. So we are now here observing the inside of the blood capillaries. Left side is naked siRNA, right side is unit PIC, and here we go. In a naked siRNA, it's instantaneously color changed to blue. However, unit PIC, it stays magenta. That means its siRNA keeps intact. And you can have a very longevity in blood circulation. So we utilize this system to treat the very intractable glioblastoma stem cells, the separate from a human patient and inoculate into the brain of mice. And we utilize antisense oligo targeting not regulated linear non-coding RNA 2G1 and by systemic injection. First, we confirm the accumulation of antisense oligo selectively to brain, as shown here, and here is a the knockdown. So there the tumor volume significantly reduced for the mice treated with this unit PIC loaded with antisense 2G1, and this is the silencing of target gene. It's more than 90% targeting occurs, and there's a significant decrease in the size of tumor. And what we surprised is that after this treatment, 100% survival of mice over 100 days for this very simple carrier systems. So if you are a chemist, you may know the characteristics of polyion complex. So of course, in a physiological condition, it's quite difficult to separate anion and cation. However, if there is some excess amount of polymer, it's easy to exchange like this way. So you can see the situation in this movie. So always there's a change in the partners. So question is that really this kind of dynamic pairing occurs under the blood circulation. So to confirm this, we again, the use FRET. So in this case, we label the polymer with donor fluorescence and siRNA with acceptor fluorescence. So first we, inject free polymer into mice. So because there should be no FRET, so color should be blue. So you can see blue color of capillaries. Then after 5 minutes, we inject free siRNA. And if this polymer catch siRNA under blood circulation, we should observe high FRET color of magenta. And actually, we observe magenta color. And for the proof of concept, we further inject non-labeled polymer to reduce the FRET efficacy, and then it should decrease to green color, and actually it turns green. So this result clearly shows the circulating polymer can directly catch injected naked siRNA in the bloodstream to form the unit PIC in situ. So this is just like a siRNA rendezvous with polymer, like a spaceship in outer space. But this is much difficult than spaceship rendezvous in outer space, because we know outer space is totally empty, but in the blood there are many the molecules and cells and etc. So this is really the rendezvous in a moving crowd. So you see the guy with red hat and red cap, and they recognize each other even in the crowd and changing their partners. So that is a kind of the very new concept of drug delivery. So you may wonder, we are utilizing the the Pegasus polymer with high molecular weight of PEG. It's a molecular weight is total 80K. So you cannot expect smooth the kidney excretion. So you may be wonder, what about the safety of remaining in the body? So we confirm that this two-armed PEG can smoothly excrete from liver by viral excretion. So here, this is the bile. So you see, this is a liver parenchymal cells, and the black region is capillary. So you can see here is a bile tracer, green, and this is a polymer, magenta. And you see the co-localization of these two colors. That means that this two-armed PEG smoothly excrete from liver bile. So this is very safe. And actually, the detail of this result is published in 2 years ago in Science Advances. And you see here, the first author is, our Indian colleague, Anjan. I think he is now attending this conference. I see his name in the audience. So anyway, please ask him about the detail. So now, this, the unit PIC system is now in a phase 1 clinical trial for treatment of recurrent breast cancer. And now, as for the next, the system to be in the clinics is treatment of glioblastoma. So what would be the future? I think that the future would be drug delivery to inside drug production. So I have no time to speak about detail, but now by utilizing nanomedicine, we can express the produce proteins from messenger RNA or plasmid DNA in desired site in the body, and also if you load the catalysis enzyme, you can even produce active drug inside of the cancer. So you can see here, the green is produced drug inside of the cancer. And then finally, we can make a system to have a gene repair by nanosystems. And actually, we, by utilizing our polymer, this is a collaboration with Neela Massey and Jennifer Dudula who discovered he got a Nobel Prize for the CRISPR-Cas9, the gene editing. So by utilizing our system, they succeed to efficiently correct the DNA mutation that cause Duchenne muscular dystrophy in mice with minimum off-target DNA damages. So in future, by nanomedicine can utilize not only for the drug delivery, but also even for the gene editing and beyond the many approaches. So finally, I would like to express sincere thanks to all of my coworkers in my laboratory and funding agencies. So thank you very much for your kind attention. Thank you very much.
Thank you, Professor, for such a wonderful presentation. It's a very informative, a good, very enlightening to know the how we can just changing the size of the system, we can target such deadly diseases. And sir, we have few questions in chat. Like, there is one question. Shall I read out to you? Sir, shall I read out you the questions that we have in chats? Yes, I'll read it out for you. One question is, can we use cationic cells directly as a carrier for intravenous administration? How these particles are different than chitosan-based polymer, which are cationic polymer-based particles, but we can't give them intravenously.
Well, maybe you're asking about utilizing cationic polymer like chitosan, and want to know about the difference from chitosan and our system. So because one of the reasons is that our system is totally cationic charge is totally shielded by polyethylene glycol to avoid the non-specific interaction of a cationic polymer with the vascular wall. That is important, and especially in a two-armed PEG system, that we just utilize a very the low molecular weight cation. It's more like oligomers. So and it is totally the covered by PEG. So because of the steric hindrance, our polymer only almost no interaction with the endothelial layer of normal capillaries, but only the interact with capillary at sinusoidal wall of liver. But as I mentioned, it does not stay in a sinusoid. It's slowly translocate into bile and excrete from our body. So that is very stable. And actually, this UNTPS system has, as I mentioned, it's already in a clinical trial and now utilizing for patients, but we have no adverse effects due to the polymer. So that means this system looks like very, very, the safe. So return to your question, of course chitosan is a very interesting polymer, but problem is that it also shows a very strong interaction with blood compartments. So you need some strategy to reduce the non-specific interaction of chitosan with blood compartment, the blood components and vascular wall. Is that okay for your is that answer for your question?
Sonali, have you any other questions, or shall we move forward? Okay. So yes, shall we move forward? We have another question from Ali, that did you observe any side effect on normal cells when you were modulating the size of the complex to avoid heterogeneity of EPR?
You mean, okay, the side effects. And as for the, the mice loaded with anti-cancer drugs, of course, you can reduce the non-specific accumulation to healthy tissue, but of course, you cannot make zero. So still, we observe some side effects, but what we can say is that we can significantly reduce the side effects due to cytotoxic reagents. And according to our experience, there seems to be no particular side effects due to the polymer itself. So we can say that side effect due to polymer looks like very very moderate or not so strong. But still, the cytotoxic regions, of course, you can control the distribution, but still there are some non-specific accumulation to healthy tissues, of course, and it causes some side effects, but not serious.
So we have another question from Ali, that what could be the reason of memory T cell retention in complex treated mice?
Could you say it again?
Yeah. What could be the reason of memory T cell retention in complex treated mice?
That is that is not bad due to the delivery systems. It's because of the immune checkpoint. So as I mentioned, if immune checkpoint inhibitor works, it recruits the cytotoxic T cells, and eventually once the cytotoxic T cells are recruited, the cytotoxic T cells are the become memory effect. So they have a memory. They remember the antigen. So the cancer cell with same antigen, the have a recurrency, these cytotoxic T cells in the second time immediately attack these cells to prevent the cancer recurrence. So the memory effect is due to the antibody itself.
So we have another question. Again from Ronali, the polymeric micelles having OH group but having positive zeta potential, will it give the sheath effect or reduce blood interaction or macrophagic uptake?
Well, so you are talking about the polymeric micelles, as I mentioned. Of course, core has some positive zeta potential, but if you can make system to be totally surrounded by PEG with with high molecular weight like a 10K, then almost you can shield the zeta potential actually. So that means you can significantly reduce the non-specific ionic interaction, and eventually you can reduce the non-specific uptake by macrophage. But if you like to make system to for example, you want to activate macrophage, then you can make system, for example, at a tumor microenvironment, maybe you can design your system to liberate the PEG code, PEG palisade. And then the system is quite available for macrophage. So it depends. So you want to make a whole system totally sterile for all the process. You can make very stable configuration. But if you want to make system to be the non very sterile only under the blood circulation and become the easily recognized by target cells, you can make system to liberate the PG to make system smart, just like a anti-PD-L1 antibody which we prepared.
Okay, thank you, Professor. I think there are no more questions from the audience, so we can move on to the next part of our session or the next speaker.
Okay, thank you very much.
Thank you. So I'll just share the introduction for the next speaker, Professor Katti.
Ladies and gentlemen, our next session is about to begin. May I request you to kindly mute your microphones and wait as we prepare for the session. Thank you.
Ladies and gentlemen, we are delighted to introduce Professor Dhirendra Khatti, who joins us from Indian Institute of Technology, Kanpur. Professor Dhirendra Khatti is a PhD in chemistry from Bombay University. Dr. Khatti was a postdoctoral fellow at Department of Chemical Engineering, Drexel University, Philadelphia, USA, and subsequently a research assistant professor at the same department. He then became an assistant professor, tenure track, at the Department of Orthopedic Surgery and the Department of Biomedical Engineering at the University of Virginia, Charlottesville, USA. He returned to India in 2004 as an assistant professor at Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. He was also the head of BSP department from 2016 to 2019 and is currently a chair professor at the BSP department, IIT Kanpur. His research interests are in the area of biomaterials, drug delivery systems, tissue engineering, and nanobiotechnology. Ladies and gentlemen, please join me in welcoming Dr. Dhirendra Khatti.
Yes, I'll request Professor Dhirendra Khatti to take over and share his screen and start the presentation.
Well, the system is not letting me share.
Oh, okay, sir. We are looking into it.
So could you please try once more?
Okay. Can you see my screen?
Not yet. We are looking into it, sir. Please give us some time. So it's visible now.
Your screen is visible.
It's saying video isn't working for me.
So your screen is visible now.
I cannot see my screen. Do you see my slides?
So, yes, we can see your slides.
Okay, so am I, is the first slide visible where it says vaccine, nano-vaccine
Yes.
for multi?
Yes, it's visible.
Okay, and is my pointer visible?
I could not see your pointer.
Okay.
Yes, it's visible.
Visible? Okay, so should I start?
Yes.
Okay, so very good morning.
Yes, sir, please start.
Okay, so let me start. So very good morning to all the attendees. At the outset, I want to thank the organizers for inviting me. I have had a very special relationship with Professor Rinti Banerjee in that we both approximately started at the same time as assistant professors, but in different IITs, and were colleagues for almost 17, 18 years. And we interacted a lot because of our common domains of research interest. And it is an honor to be speaking at this occasion. And the topic that I'm going to be talking on is a topic that a student of mine started here at IIT Kanpur, and he then later on went on to work with Rinti Banerjee, although not on the same topic, but related topic. So let me get started now. So my talk is on the development. So it's the story is a single story. So I'm not going to be giving you multiple examples. It is just one example that I'm walking you through. And it's titled Vaccine/Nanovaccine for Multi-Drug Resistant Diarrhea. So multi-drug resistant bacterial diarrhea is also known as shigellosis. It is the leading cause of bacterial diarrhea, which causes greater than 0.2 million deaths globally in 2016. It's caused by a bacteria called Shigella. Shigella was discovered by Professor Shiga in 1897. It's a gram-negative bacteria, foodborne pathogen, and mode of transmission is fecal-oral route, highly contagious in that only 10 bacteria are sufficient to cause the disease. It has 4 species and more than 50 serotypes. Symptoms typically are 1 to 3 days post-injection. You have abdominal cramps, tenesmus, diarrhea, which is also can be bloody, fever, vomiting, nausea, and can last up to 7 days. Complications, if not treated well, it can lead to these complications. Dehydration, toxic megacolon, hemolytic uremia syndrome. This is typically there only if this Shigella dysentery, which has the Shiga toxin in it, is exposed, the patient is exposed to Shiga toxin. Then rectal prolapse, seizures, especially in children, sepsis, and even death in neonates and malnutrition children. Long-term effects can normally diarrhea does not have, but shigellosis can have. So long-term effects, stunted physical growth, reactive arthritis, and this can be multiple types of things like Ritter syndrome, 1 week post-infection, joint pain, painful urination, eye inflammation, etc. Vulnerable groups are infants and the elderly, especially in low and middle income countries or LMICs. And diagnosis is stool culture on selective media, stool staining, biochemical tests, and more recently PCR. Preventive measures are simple, but unfortunately still are not available in many parts of the world. hygiene, hand washing, and clean drinking water. And in the recent past, COVID has also told us the importance of hand washing. So with this background, I will go ahead into what's the treatment. Treatment is pretty simple. It's fluids or electrolytes, zinc tablets, and antibiotics. So most often, if treated well and if the infection is not severe, most patients would sort of survive and there would be any problem. It's just like a bout of diarrhea that comes and goes. But the complications have arisen primarily because this bacteria has now become antibiotic resistant. And this antibiotic resistance has sort of curtailed our ability to treat it. Now I'll give you some statistics, global burden. I just mentioned greater than 0.2 million deaths in 2016. And if you see this world map, you can see here the red regions, Southeast Asia, including India, Nepal, Bangladesh, Pakistan, and then Southeast Asian islands, Malaysia, Indonesia, etc. Then the entire South Africa. These regions have got a higher prevalence and higher death. So you can see here that the deaths are around 1 to 76 in that range for all these per lakh. And 1 lakh is a fairly, or 100,000 is it seems like a fairly small number, but when you multiply it for the population, it's a very large number. Higher morbidity in low and middle income countries due to poor water quality and sanitation, and this is the main problem. Now in India, we have had multiple times that it has come about. It was endemic, and in India, we have had outbreaks multiple times in West Bengal, multiple times in Kerala. Most recent one was in 2020 in Kerala. All Shigella outbreaks have been caused by multi-drug resistant Shigella. So what is done is, after the outbreak, they take clinical isolates and they test them, and then they test if they are multi-drug resistance or not. And that's how we come to know that whatever outbreak is there is because of a multi-drug resistant variant. So, and these multi-drug resistance are all resistant to the common antibiotics. Now, the solution typically is a vaccine. And vaccine, as you know, one normally takes, works with the pathogen, components of the pathogen, or you generate an antigen, and along with an adjuvant, you can activate the innate immunity and then eventually adaptive immunity, and as a consequence, allow the body to protect itself from any such bacterial attack. Now, however, antigens from one species or serotype may not protect against another, as Shigella has more than 50 such serotypes and sub-serotypes. A licensed pan-Shigella vaccine is still not available. So what are the strategies for vaccine development? Just in the interest of understanding, we can have whole cell vaccine or subunit vaccine, and whole cell vaccine can be live attenuated or it can be killed inactivated. The subunit vaccines can be components of the pathogens, as the name suggests. And now, more recently, we have nucleic acid based vaccines, which is basically things that work with DNA, RNA encoding antigens. Now, there are multiple advantages and disadvantages associated with each of these techniques. And just to quickly go over the advantage, in the whole cell, actual pathogen does excellent stimulation of the immune system, but there is a risk of disease. In the subunit, only protective antigen use, so low risk of adverse reaction and less possibility of disease, but may require repeated doses and an adjuvant. In nucleic acid, long-term immune response and vaccine stability, no adverse reaction, but it's relatively new and few in number. So these are the advantages and disadvantages of these systems. But in spite of all these strategies being available, the current problem of Shigella is that a commercial Shigella vaccine is not available due to low immunogenicity or serotype specific protection, i.e. there is no cross protection possible. Now, what is cross protection? So cross protection is something that is allowing for one antigen to work against multiple of these Shigella types. So all the serotypes, if you can sort of nullify every one of them using a single antigen, then that antigen would be referred to as a cross-protective antigen. This is normally very difficult to come across unless it is a conserved antigen throughout all the species. So therefore, conserved immunogenic proteins found in Shigella would be good for cross-protectivity. There is something known as the type 3 secretion system in this bacteria. Now here I'm showing you this. On the bottom is the bacterial cell, and on the top is a host cell or a target cell that the bacteria is trying to infect. Now typically it infects in the gut, so hence this is the gut lining and this is the Shigella bacteria. Now this little bar or cylindrical shaped structure that you're seeing here, this orange spheres followed by red spheres followed by these green spheres, this is referred to as an injection system, and more specifically the type 3 injection system. At the tip of this injection system, at the point where it is infecting the host cell, we have these invasive plasmid antigen proteins, which are referred to as IPA proteins or IPA B, C, and D. And these proteins are conserved between all the serotypes of Shigella and overcome the challenge of serotype-specific immunities. So IPA B and C are found to be the most immunogenic of these. And IPA B has shown independent vaccine efficacy, and IPA C has limited reports because it's an unstable protein and therefore undergoes degradation when it's used. So relatively B is more stable, and therefore B has been explored independently. So therefore, if one can use any of these conserved proteins, then it sounds like a good option. Therefore, conserved proteins can be a target for cross-protective vaccines. However, instability of these immunogenic proteins such as IPA-C aggravates the problem. So before we get into our data, the typical attributes of a good vaccine would be: it should be safe, it should be immunogenic, should protect against all Shigella. That means you should have cross protection, and should be cost effective and amenable for mass immunization. Non-invasive route is preferred because it is the easiest way to administer. So oral or nasal are the two non-invasive route, and within those, intranasal immunization is preferred because it has better immune response, no acidic environment like the stomach for oral, a thin mucus layer, and dosage minimization and cost minimization is there. So therefore, nasal route is a preferred route. Now, based on all this literature that I just showed you, we decided to work on a conserved antigen IPA-C, although it was unstable, and then try and stabilize it and characterize it and test it as an intranasal vaccine. This was our first set of experiments. So we took the route of stabilization. Now IPA-C, if you see what I'm showing here is IPA-C derived from Shigella dysentery or SD. This IPA-C has a molecular weight of around 42 kilodaltons, and we had tried to stabilize this protein, and it turned out that in the interest of time, I'm going a little fast. It turned out that the purified IPA-C was unstable and it degraded within hours. So this band that you're seeing here of purified IPA-C, which is immediately post purification, this band disappears after a few hours. So the same purified IPA-C, if stored and then run on the gel again, the band disappears, suggesting that it has degraded. So it was unstable. Now, previous literature showed that if you take the cell lysate when you're purifying the protein that is expressed, and it was sort of treated with this surfactant LDAO, then this LDAO treatment or OPOE, LDAO is lauryl dimethylamine oxide and OPOE is octyl polyoxyethylene. Both of them sort of maintained the protein structure. If you see this clearly, there's a proper maintenance, whereas against when used in presence of urea, the protein sort of starts undergoing denaturation. So this suggested that maybe this treatment with OPOE or LDAO can help. And this again is showing stability data when used as is. These are days 1, 2, 3, 4, 5, 6. It can be stable up to 2 days and then it degrades. When used in presence of urea, also it can be present up to 2 days and then it degrades. When present in LDAO, you see it goes on till 6 days. OPOE, it goes on at least for 4 days and then starts degrading. So relatively amongst all these, LDAO was the most preferred in terms of stabilization. So stability can be increased with LDAO is what we found out. And therefore, then we decided to work with LDAO and stabilize. And again, in the interest of time, I'm going a little fast, but I'll draw your attention directly to this part 9 and 10, columns 9 and 10, where we have used 0.05% of LDAO. And here, this quantification of the same. So you can see here, compared to all the other lanes which used various concentrations of LDAO, the one which used 0.05% showed the highest extraction of the protein, and the protein was stable. So therefore, this system, which was almost twofold when compared to untreated systems, was then optimized concentration is what we used. So LDAO increases solubility and yield of IPAC, and 0.05% is the optimal percentage. We then developed 3 processes, and all the 3 processes we showed that when you use LDAO appropriately in the soluble fraction as well as the insoluble fraction for the extraction of the protein, it turned out that LDAO was stabilizing the protein and we were getting a good percentage yield. And you can see here in this gel that the pooled one, which consists of both the soluble and the insoluble portion, is a big thick band. So that's a reasonably good product. And here is sort of a summary of the 3 processes. You can see that IPA stabilization protocol, and wherever I'm showing LDO in red is where we have inserted the LDO for the purpose of stabilizing IPAC, which was otherwise unstable. And what I'm showing here on this slide is for a 4-liter culture, which leads to a 10 gram of E. coli pellet. And so this was leading to a total of this 6.8, 37, and 34 milligrams. So you can see that each process is sort of improving the amount of yield. So, so far, what I've shown is that we had a good conserved antigen, IPAC. The IPAC was highly unstable, and using this interesting molecule LDAO, we were able to sort of extract the protein in large quantities and stabilize it. Now, going forward, we wanted to characterize this protein, and we wanted to check if it was aggregating. Now, you see here on the right-hand side, I'm showing you the protein after centrifugation. And normally, it is known to be aggregated and it's unstable. Therefore, it undergoes aggregation. But with the current protocols that we developed, not only did IPAC not aggregate, but also maintained the secondary structure. And this graph below is showing you the maintenance of the secondary structure. The blue one, or the cyan color, shows the soluble fraction. And then the maroon color is the soluble and the insoluble fraction. And you can see that the secondary structure of the protein is clearly maintained. Similarly, the lyophilized protein as well, before lyophilization and after lyophilization, there was no change in secondary structure, suggesting that this protein was stable. And Western blotting stored at IPS at 4 degrees Celsius also showed that there was no problem. This is a transmission electron micrograph image of the protein, and you can see that it is sort of like a spherical aggregate. Now, temporal stability is what we wanted to check. So in this slide, I'm showing you at 5 temperatures, -80, -20, 4 degrees, 25 degrees, and 37 degrees. So 37 degrees is physiological temperature, 25 is room temperature, 4 degrees is the regular 4-degree refrigerator, which is used most often to store vaccines in the clinic or anywhere primary healthcare center. And in more tertiary care centers, which are more sophisticated, you may even have a -20 and a -80 degrees refrigerator. It turned out that at lower temperatures, this protein was stable for longer durations. -80, 14 months. -20, 14 months. 4 degrees, 12.4 months. 25 degrees, 1.25 months. And 37 degrees, it was only for a day or two. So which means that as the temperature is increased, it would keep decreasing its stability. But it's important to note that at 25 degrees Celsius, it was able to stay for 1.25 months, which is close to 45 days. And at 37 degrees, you could treat it in the human, in the body, and it would be fine for a day or two, which means its effect would be there in place. You can also, it's important to note here that slightly here where it's depicted DP, that is the small band which is coming because of the degraded protein. That means the degradation process has begun, but we still have a proper band here at 42 kilodalton. So this was the study of the temporal stability, and this is a summary of the same. When compared to reported stabilities, we found that the stabilities that we obtained using LDO were far higher. So this was a few weeks here at -80. We were at 14 months at -20. There was no data that we could find, but we were at 14 months there as well. 4 degrees Celsius, it was less than 2 weeks. We were at 12.4 months, and room temperature, it was 6 days. That was reported for 21 degrees. We were at 25 degrees, and we could go for almost 40 days. So these, and 37 degrees, there was no data in literature, and we've shown at least 1 or 2 days of proper stability. So this sort of now made this protein or antigen stable at room temperature, and that was good because another important point I should mention along the side here is most antigens or most vaccines require a cold chain. And this cold chain requirement is sort of circumvented if you have a temperature stable antigen. So this was another important thing. And it's important primarily in the context of low-income countries where cold chain is probably not there in many remote rural areas. So the next experiment we did was to test the immunogenicity of this stabilized iPSC. So here I'm showing you, we gave the intranasal immunization was the route of immunization. We gave 3 doses, first day, 14th day, and 28th day. And then we kept a gap of 14 days. And on 56th day, we gave an intraperitoneal challenge using heterologous Shigella. And then 14 days later, we terminated the study. So now I'll show you data. So after 28th day, which is when the 3rd dose was completed, we took the sera to check if it can produce antibodies against Shigella. And then we saw that both for the Shigella dysentery serotype 1 as well as Shigella flexneri serotype 2A, you can see the bands, which means that iPSC was derived from Shigella dysentery, was able to do what we were expecting, cross protection. Here at the bottom, I'm showing you IgG and IgA, and you can see that the IgG and IgA levels went up till the 14th day and remained high up to the 45th day of study. And here we're showing you, I'm showing the cytokine concentrations of IL-6, IL-10, and IFN gamma. So you can see the IL-6 and IL-10 picked up as we increased the dose of iPSC from 10 to 20 to 30. And IFN gamma, although on this relatively it looks small, but it also was increasing continuously as we increased the dose from 10 to 20 to 30. So therefore, iPSC immunized sera recognized a heterologous species, the first important point. This is enabling cross-protection and resulting in significant antibody response and a dose-dependent cytokine response as well. We then looked at the protective efficacy. So after having established the immunogenicity, then we looked at the protective efficacy. And in this protective efficacy, we have used a high dose of Shigella flexneri 1 × 10⁹ CFU. In this gross images I'm showing you 4 hours post-challenge. In the control, we had visible diarrhea, whereas in the vaccinated group, there was no diarrhea. In some of the controls, we also had bloody diarrhea, which is known if you are infected with Shigella dysentery. Then the intestine, when it was homogenized and it was colonized, we found that the intestinal homogenate cultured led to a colonization of Shigella in the control. And in the vaccinated group, there was no such culture. Then further, weight loss, we saw that in the vaccinated groups, particularly slightly higher doses, 20 and 30, there was very minimal weight loss for Shigella dysentery, whereas for Shigella flexneri, relatively almost no weight loss. And when we checked at the survival, we saw that all the groups, 10, 20, and 30, survived more than 20 hours. But if you observe carefully, these two groups, which is 20 micrograms and 30 micrograms, The maroon color is 20 micrograms and the blue color is 30 micrograms. In Shigella dysentery challenge of 10 to the power 9 survived, whereas all other groups did not. Up to a maximum of 36 hours they survived. And in the Shigella flexinary challenge, both the 20 microgram, 30 micrograms were 100% survival was seen. So therefore, iPSC showed cross protection and both sets were protected from diarrhea, colonization and weight loss without any antigen. So, adjuvant. So this is important to note because we're just giving an antigen and nothing else. So major factors. So now that we had, we had sort of stabilized and established iPSC can work as an antigen. So then we wanted to improve its translatability. So for that, we looked at the major factors for translation. So one is we need a maximum protection in minimum dose. Second, we want to reduce hospital visits for patients. And the third, we want a non-invasive route like the intranasal route, which can even together lead to mass immunization possibilities. So we decided to work with vaccine delivery systems, nanoparticles, which are known to be amenable for mass immunization. So we wanted to minimize the requirements of a booster dose. We wanted a protective antigen against degradation, and that was already there in our hand now, iPSC. We were looking for high payload delivery, adjuvant effect coming from this nanoparticle is what was known in literature, and biodegradable with sustained and tunable release kinetics. Now, amongst the system, interestingly, there is an FDA-approved polymer, polylactide co-glycolide, which many of you may be familiar with. It is interesting, while it is very popular in terms of drug delivery systems, for Shigella, somehow there have been only 2 systems that have been developed for vaccine delivery systems, or attempted, I should say. One is polyanhydride and the other one is chitosan based. And in both these cases, the protein was 40% and the nanoparticle protein gave around 67% survival. So this, if you remember the data from the previous slide, we were showing with bare protein almost more than what was possible using a nanoparticle formulation. But nonetheless, this data is indicating that you can go from bare protein 40% to nanoparticle protein to around 67%, which means you can get a 25-30% increase using a nanoparticle system. So therefore, vaccine delivery systems can increase immunogenicity. So this is what we started with, a PLGA system. Now, again, in the interest of time, I won't get into this technique, but we made PLGA nanoparticles of around 350 nanometers and -25 zero potential, and showed that it could encapsulate the protein of interest here, the antigen iPSC. And this is the released iPSC, 0.8 micrograms. You can see a very faint bar here. Then the release and degradation is what we studied. These nanoparticles, they released the protein over a period of 28 days, almost 80% of the protein, which is 77% of the loaded protein was released. And it followed this Fickian diffusion. This is the Course-Meyer-Pepper's plot. So it followed Fickian diffusion. And these nanoparticles, they had sort of good morphology at day 1, which slowly started fusing on day 14. And then by day 35, they had completely degraded. So we wanted a relatively faster degrading system that could degrade in 1 month. And this is what we got. So therefore, we were happy and excited about this delivery system. So slow and sustained release of protein from PLGA nanoparticles. That means over a period of 30 days, we could get approximately 80% release through a Fickian diffusion mechanism. And these nanoparticles would be completely degraded by 35 days. So the animals should be challenged after all this is over. So we had an approximate timeline for challenging. Safety and immunogenicity. So we did take the iPSC nanoparticle system with 10 microgram and 20 microgram, and we found that the LDH cytotoxicity assay, which was done when compared to the lysis buffer control, was very, very minimal, indicating that this system is safe. In this case, what we did was we had a single intranasal dose in bulbsy mice on the first day, and then we waited for 35 days based on in vitro data that the nanoparticles take around 35 days to degrade. And then we challenged either on the 43rd day or on the 56th day, and then waited for 14 days, which is on the 57th day or the 70th day, and terminated the study further. So first was, of course, here again, we saw the immunoblots. Both the species were visible. Here again, if you see, in control, it's almost on the x-axis, whereas the vaccinated group shows a very high IgG count. Similarly, an IgA count, control is on the x-axis. Here again, quantification of the cytokine shows that IL-6, IL-10 and IFN gamma, just as we saw in the free protein case. Here 10, 20 and 30 iPSC concept, this is the free protein. This is the single dose iPSC protein, which showed a relatively higher IL-6, IL-10 as well as a comparable IFN gamma. This was 30, which was slightly higher than the nanoparticle one. So these, now based on this, I should tell you that the comparisons are going to be made with iPSC 20 and this. The reason is because the dose used in the nanoparticle is 20 micrograms. So a fair comparison would be between these 3 bars and these 3 bars. And if you compare those, then the nanoparticle system is showing higher bars, both for, I mean, all for IL-6, IL-10, as well as IFN gamma. And this is the quantification of the fold chain. And so that's showing a pretty good data. Now, protective efficacy. So here again, 4 hours post-challenge, control, you can see there is diarrhea. Single dose, it's a proper pellet, fecal pellet, so no diarrhea. Here, weight loss, very minimal with the nano vaccine. This is 16 hours post-challenge. These mice, you can see, they have diarrhea and they are sort of crouched and sitting in one corner. These are the ones which are the control mice. They have diarrhea, visible diarrhea. Whereas if you go to the group which is vaccinated, you can see they're much agile, difficult to catch. They're running around and then they don't have any visible diarrhea. So this system is working as a vaccine delivery system. And if you look at the survival plot, you can see here single dose. The control mice all were dying at around 36 hours, whereas the single dose had around 60% survival up to the 70th day time point. So single dose nanovaccines protected immunized animals from diarrhea, weight loss, and lethargy with approximately 67% survival. Now, while we had made this system, and it was very exciting, we wanted to further improve this system. And it was known that in literature that there are these biomimetic nanovaccines where one can increase the T helper 1 response, which can sort of enhance the immunogenicity of the system. So increased Th1 response is beneficial for elimination of intracellular pathogens such as Shigella. It was known, and involving multiple immunostimulants have been reported to increase Th1 response. So we wanted to have these immunostimulants. And 2 of the known immunostimulants are LPS and CpG DNA. Both in human and mice, they have been found to be increasing Th1 response. So in one previously reported study where they took latex beads and they had this iPSC antibody on which they put the Shigella culture, and then you can see here in low and high magnification that there are multiple particles which are taken up by the cell because of the presence of this iPSC and Shigella on top. And this is just showing how the ruffling of the membrane takes place when in the presence of that system, which is not happening when iPSC is absent, suggesting that iPSC is facilitating the process of entry. So now surface presence of certain moieties could increase immunogenicity and uptake. So basically surface presence of this CpG DNA or iPSC would sort of increase. That was our hypothesis. And so we decided to work with them. Now for that, we made different types of nanovaccines. And in the interest of time, going a little fast, but I'll show you. We had made 3 vaccines. We termed them Nanovaccine 1, 2, and 3. In Nanovaccine 1, we had the same as the previous nanovaccine, that is iPSC inside PLGA nanoparticle. But we had iPSC, these gray dots on top. So it was decorated with iPSC on top, which was going to facilitate T helper 1 response and hence improve immunogenicity. In Nanovaccine 2, we had the same system, but in addition, we had CpG DNA, which is another immunostimulant. So we had now a second immunostimulant. And in Nanovaccine 3, we had IPAC, this system same as NV2, but in addition, we had IPA-B and LPS, LPS being another stimulant and IPA-B being another IPA protein, which is conserved. So here, this table shows the 3 vaccines clearly. NV1 or Nanovaccine 1 has IPAC as the antigen and nothing else. NV2 has 1 antigen and 1 immunostimulant, CpG DNA. And NV3 has got two antigens, IPA-C, IPA-B, and has two immunostimulants, CpG-DNA and LPS. So this is how we increase the order of complexity from NV1 to NV2 and NV3, and we got three types of nanovaccine systems, and we wanted to study if these systems can do any better. So first thing was to get these antigens and immunostimulation. So again, in the interest of time, I'm going to go a little fast. So we were able to get LPS isolated, and then we were able to get IPA-B and IPA-C and formulate them into these nanoparticles. And here I'm showing you nanoparticle characterization data. This is prior to surface modification. So they were around 250 to 300 nanometers in size, had around -20 to -25 zeta potential. The released antigens were all fine. And the FTIR characterization showed that we were able to get the surface modification done on top. This surface modification, certified modified nanovaccines, which were referred to as NV1, NV2, and NV3, were re-characterized for their size and zeta potential, and were then studied for their cytotoxicity using LDH assay. And in cytotoxicity, all the three nanovaccines were found to be non-cytotoxic, and this is with respect to the control lysis buffer. Then we studied in vitro uptake of these nanoparticles. We wanted to know if the presence of these immunostimulants on the top, as has been reported, does it increase the uptake? I don't know if these little red dots are visible, but I have the quantification here of these red dots, which are TRITC dye labeled nanoparticles, and this is the DAPI staining. And so here you have NP plus IPAC, which is NV1, and then this is CpG-DNA plus IPAC, which is NV2. So if you look at them, you can see here that compared to control, the presence of the surface modified increases the amount of uptake into these nanovaccines into the cells. So therefore, presence of IPAC can increase cellular uptake. Then immunogenicity. When we studied the immunogenicity, we had, just as previously, 3 doses, day 1, day 14, day 28 by intranasal immunization. And then on day 56, we're leaving a gap between 28 to 56. And then we did an intraperitoneal challenge, then let go another 14 days, and then did the termination of the study. Here again, as I've shown previously, we got very good data in terms of IgG as well as IgA. And if you look at IL-6, IL-10, IF, and gamma, you can see here in IL-6, NV1 was high here and NV2 and 3 were a little lower. But if you look at IL-10, it was a continuous increase with the increase in complexity. Similarly, IF and gamma, there was a continuous increase in complexity from NV1 to NV2 to NV3. And it was sort of free proteins plus LPS was our positive control. And you can see that free protein plus LPS was also doing fairly well. When we look at the challenge, you can see 4 days post-challenge. Control, there was visible diarrhea. And vaccinated, there was pellet, so there was no diarrhea. Weight loss, all the systems did not show any weight loss compared to control. And survival, all the vaccine systems, NV1, 2, 3, showed much better survival compared to the untreated control. Even the free protein showed a fairly good survival. Now, this last part is something which is very interesting. So we wanted to see if we can get the progeny to be protected if the parents are protected. So here what we did was we took the parents and vaccinated them with the simplest vaccine, which is the NV1, IPAC coating on top and inside. And then we had 3 doses. And then after the 3rd dose was given, we allowed for mating. Then the neonates, we took the neonates, and then on the 5th day, we challenged the neonate orally with Shigella. And we found that the neonates were also protected. That is, we were able to get passive protection in the progeny. Here I'm showing the unprotected progeny, which could survive up to around 24 hours, whereas the protected progeny were fine, up to around 85% survived without any problem. This was, mind you, the simplest vaccine. So therefore, right till now, what I showed you was that all the vaccines, the complicated vaccines that we developed, all the vaccines were cross-protective. NV1 can be explored if the cost is a constraint, which can be a constraint in countries such as India and Africa. And if not, both NV2 and NV3, which will be relatively more expensive, can also be used. So overall, this is the summary of the study. In part 1, in the first part where we developed just the iPSC protein antigen stabilization, it is known to be unstable. We stabilized it with 3 processes, increased the yield, concentration, and temporal stability. It showed self-adjuvanting effect, and we got a cross protection of almost 100%. In part 2, we developed a single dose nanovaccine. We had a minimum protective dose. Based on part 1, we chose that minimum protective dose as 20 micrograms. 77% release was there from these nanoparticles in 28th day. All the nanoparticles degraded at 35th day. We had a high cytokine response, which was 3x of iPSC alone. And the cross protection was around 67% survival. In part 3, which is the biomimetic vaccines, which is surface modified nano vaccine delivery systems, Shigella antigens and immunostimulants were encapsulated. Surface functionalization was done for these nano vaccines, results in higher cellular uptake, cross protection of all the vaccines, approximately 80% survival in NV1 and NV3, and passive protection, which is a big highlight. That is, the progeny are also protected if the parents are protected. Passive protection is approximately 90% survival against heterologous species. So before closing, I would like to thank my funding agency. DBT has been very generous. Department of Biotechnology. I've been supported almost for the last 9 to 10 years for this project. And this work was initiated by Vinod Patil, done by Nadeem Ahmed, who was working with Rinti Banerjee until recently. And all the data that I presented today was largely done by this student, Namrata Barua, who is about to graduate and is looking for a postdoctoral opportunity if you're interested. And I want to thank my collaborator, Hemant Kole, who is at National Institute of Cholera and Enteric Diseases, where some of the studies were conducted. And namaste, and thank you all for your attention.
Thank you, sir, for such a wonderful presentation of the work and giving us insights into this in this area, what things are being developed. So I now request the audience to have some questions.